session, but as you have observed that before lunch we have felicitated our esteemed guest and senior members of Society for Radiation Research. So to complete that important ceremony, uh, I would like to request Dr. J.K. Singh to felicitate Dr. Nagraj Hulgol, who was our first president of Society for Radiation Research. He was the highly inspiring personality for young radiation researchers, actually, and I must say SRR actually is evolved from the mind of Dr. Hulgol and Dr. K.P. Mishra. So it is that way, and please, and also I will request and welcome Dr. Sudhir Chandana also from INMAS. Dr. Sudhir Chandana, please come on to the dais. Yes, please come. And Dr. J.K. Singh will felicitate both the senior scientists.
We express our gratitude for his exceptional and truly outstanding contribution in radiation research, and for this we would like to give the Lifetime Achievement Award to Dr. Nagraj Hulgul sir. He is an eminent name in the area of radiation therapy and research. He is the immediate past president of SRR India, and currently he is an editor-in-chief of Journal of Radiation and Cancer Research. After graduating from Dharwad University, Karnataka in 1975, he decided to take up radiation oncology for his post-graduation at Aiims, Delhi in 1980. He then joined TMC in Mumbai as a registrar, and he is a recipient of many national and international journals. So I request J.K. Singh sir to give away.
Yeah, to participate in this important ceremony, may I request Dr. K.P. Mishra, the founder president of Society for Radiation Research, so that we can begin with this.
I also request Dr. Sudhir Chandran to please come on this stage. VK Singh sir to please felicitate Dr. Sudhir Chandran, Senior Consultant Radiobiologist. Thank you, sir. Now I hand over mic to Dr. Ayushi for continuation of the next session.
Moving ahead with the event, next on board. Okay, plenary is not to be conducted. We move further on the event. The next event would be SRR Young Scientist Award presentation, and I invite Dr. Vijay to start off with the session.
Good afternoon. So without losing any time, we should start the session. So first Young Scientist Award candidate is Saurav Kumar Das. There will be 8 minutes time for presentation and 2 minutes time for question and answer.
Okay. So, good afternoon everyone. It's a great pleasure to be here today. Myself Saurabh Kumar Das from Radiation Biology and Health Science Division, BRC Mumbai. The topic of my presentation is mechanistic insight into radiobiological response of lung cells to thorium in vitro and in vivo investigation. First of all, as we all know that thorium is a potential fuel material in DAE's three-stage nuclear power program, in which thorium is going to be utilized in stage 2 and stage 3 nuclear power reactors. And the recent developments in thorium fuel suggest that thorium can be utilized in existing nuclear power reactors like PHWR and CANDU reactors. India is utilizing thousands of tons of thorium per year in different IREL sites. So during the process of mining, milling, thorium purification, fuel fabrication, fuel reprocessing and waste disposal, there is a chance of contamination of thorium to the occupational workers through several routes of exposure. Among these routes, inhalation is the prime route of exposure, which contributes for more than 85% of the total amount of exposure, and lung is the primary target organ. So as I told you that lung is the primary target organ for thorium under inhalation condition. This is the structure of alveoli, which composed of alveolar epithelial cell type 1 and type 2. These type 1 cells are thin cells responsible for gaseous exchange function in lung, and type 2 cells are responsible for production of surfactant proteins. These surfactant proteins are responsible for decreasing the surface tension during respiration to prevent the collapse of alveoli. So we have studied the effect of two different chemical form of thorium, thorium nitrate and thorium dioxide in alveolar epithelial type 1 cells, and results suggest that thorium dioxide internalizes into lung cells through receptor mediated endocytosis, but thorium nitrate internalizes through membrane perforation. Although both the chemical form of thorium induces ROS generation, but it is the proteotoxic stress which is responsible for inducing the cytotoxicity in lung cells. Coming to the mechanism of thorium-induced cytotoxicity, despite of their differential internalization, thorium nitrate and thorium dioxide, they both produce ROS through oxidative stress, causes proteotoxic stress as well as DNA damage response. These all together are responsible for thorium mediated cytotoxicity. And as I have told that type 2 cells are responsible for production of surfactant proteins. There are 4 types of surfactant proteins: A, B, C and D. A and D are responsible for immune defense function, but B and C are responsible for decreasing the surface tension. So we have studied the effect of thorium on the surfactant proteins, and results suggest that after thorium treatment, SPD is found to be upregulated in type 2 cells but downregulated in type 1 cells. But other surfactant proteins are found to be downregulated after thorium treatment in both type 1 and type 2 cells. To further study the nuclear localization and cellular internalization of thorium-induced surfactant proteins, we have performed immunofluorescence analysis, and results suggest that SPD is found to be nuclear localized in type 2 cells, but other surfactant proteins are not getting localized after thorium treatment. And pre-treatment with the inhibitor of ROS, HSP90 and ATM, that is DNA DDR response protein, are responsible for down-regulating the thorium-mediated upregulation of surfactant protein D, that suggests that role of these signaling molecules in the thorium-induced upregulation of surfactant protein D. So to validate these in vitro data in mice model, in vivo model, we have exposed mice to thorium aerosols and collected the lung tissue for histopathology and Western blot analysis, and the results suggest that thorium is found to increase the expression of SPD but decrease the other surfactant protein expression. This suggests that SPD can be used as a potential biomarker for thorium mediated toxicity. So summarizing this result, thorium found to alter the expression of surfactant proteins, and there is a role of DDR, oxidative stress and proteotoxic stress in thorium mediated regulation of the surfactant proteins. So then as we have discussed that thorium is inducing ROS generation. So ROS is found to cause double stranded break in DNA. So we have studied the DDR mechanism in details after thorium treatment in lung cells, and the results suggest this immunofluorescence of gamma S2X suggests that thorium is found to induce gamma S2X, which is equivalent to 2 gray of radiation, and this is further validated in Western blot results also. Coming to the next part, we studied the effect of thorium on the DNA damage response sensor proteins like ATM, ATR, DNA PK and their phosphorylated counterparts, and we found that thorium is found to induce the expression of these sensor DNA DDR sensor proteins. Then coming next, we have studied the effect of thorium on the HR response protein, those proteins which are responsible for homologous recombination repair, and we found that thorium is found to downregulate the expression of HR proteins like RPA, MRN proteins, BRCA1 and RED51. And coming to next, we have studied the effect of thorium on NHEJ protein to study whether NHEJ is involved in thorium-induced DNA damage repair by expression of Q70, Q80 and XLF4, and surprisingly results suggest that these proteins are getting activated after thorium treatment. That means the role of NHEJ in thorium-induced DNA damage response repair. Then coming to the next study, we have also further confirmed these things by confocal analysis, which suggests that DNA PK is getting internalized into the nucleus of F549 cells after thorium treatment. To confirm these things, we have also performed cell cycle analysis, which suggests that pre-treatment with DNA PK and RED51 inhibitor along with thorium, the DNA PK inhibitor is found to increase the sub-G1 stage cell cycle, but RED51 inhibitor doesn't have any effect on the cell cycle regulation. We further confirm our hypothesis that NHEJ is truly involved in thorium-mediated DNA repair response. Then, as pneumoconiosis is one of the disease has been found, disorder has been found in Chinese thorium workers, we have proposed to study the thorium-mediated inflammatory regulation, whether there is any inflammation induced by thorium or not. So we have performed the thorium-induced expression of inflammatory cytokines, and found that thorium is found to upregulate the expression of pro-inflammatory cytokines, but downregulate the anti-inflammatory cytokines. cytokines to further study the role of NF-kappa B in the regulation of thorium-mediated inflammation. We have performed Western blotting of gamma NF-kappa B and its counterparts, and found that this NF-kappa B is getting activated after thorium treatment, which suggests the role of NF-kappa B in thorium-mediated inflammation. Further, we have validated these in vitro data in vivo models, mice model, after thorium inhalation. Yes, just last slide. This is the last slide. So this suggests that thorium is found to upregulate the expression of NF-kappa B, and this is my summary slide, which suggests that thorium induce ROS, HSP and DDR. Along with it differentially regulate the surfactant protein, which altogether responsible for inducing inflammation through NF-kappa B by producing these cytokines. And I am happy to share with you all that. So these are some of our publications which has been published out of this work, and one of our cover page has been featured in ACS Environmental Science and Technology. These are the publication in last 2 years from our lab, from this project. So thank you, sir.
Question from the audience. Since I'm one of the person scoring the candidate, I won't be asking any question. So question for candidate, please, from the audience. If there is no question, I would like to thank you.
Okay, thank you very much, sir.
On the small screen, time is displayed, so candidate may see the screen, TV screen, to manage the time, because that is also one criteria here. So next candidate, Niharika Nima Baider.
Good afternoon everyone over here. I am Niharika and I am here to present my work on assessment of cold atmospheric plasma induced DNA damage in A431 human skin carcinoma cells. So what is cold plasma? So first of all, I'll define plasma, that is the fourth state of matter, which is the gaseous molecules are all ionized. Now there are two varieties of it, that is thermal and non-thermal. Where the thermal plasma has a temperature reaching to thousands of Kelvin, cold plasma, that is a non-thermal plasma, has temperature ranging around 37 degrees Celsius. There are many forms of natural plasma as well, and when the cold plasma is given at atmospheric pressure, we call this as cold atmospheric plasma. Hence, I'm going to refer it to as CAP. So there are various components of CAP, and I'm going to focus on the most biologically relevant, that is ions and electrons and reactive species. So there are many applications of CAP, and here I'm going to talk about the cancer treatment. So why cancer? First of all, cancer is a huge global burden, and we know that there are many established therapies such as radiotherapy, chemotherapy, surgery, which do increase the survival of the patients, but they also have a downside, that is they cause the side effects. Now, which compromises the life of the patients. Hence, there arises a need to study various new therapies. Like there are many adjuvant therapies that we are using in conjunction with the existing therapies. Like in the morning, we heard about the hypothermia. Cold atmospheric plasma, that is CAP, is one such therapy, which is a fast-growing therapy. But the mechanism of cancer killing is yet unknown. Here arises my objective, that is to understand the biological responses of human cancer cell to CAP. So we have an indigenously developed CAP instrument in a lab that was designed and fabricated in collaboration with IPR Gandhinagar. So we have this chamber where the gas passes and is being ionized in the presence of current, which generates the arc plasma, that is the electrical plasma. This is the schematic of the probe. This is the instrument that we have in our lab. So there are various parameters by which we can determine what is the dose delivered to the biological systems. we keep the first three constant, that is voltage, flow rate, distance. Hence, we have the exposure time measured in seconds, which determines that the dose delivered to the biological systems. The best part about the system that we use is that it works at ambient temperature, it requires no special shielding, it requires minimal infrastructure, and finally it fulfills the Made in India mandate of DAE. So we went on to study the cell proliferation and cell survival, and we saw that with the MTT assay, there was a dose-dependent decrease of the proliferation almost 3 times when we compared it to the IR. Similarly, when we went on to study the gold standard clonogenic assay, we could see that again there's a dose-dependent decrease, and hence we can conclude with this that CAP has a greater tumor killing potential than IR. Now the biggest advantage of CAP has that it had a selective action only on cancer cells. Here we can see that human keratinocytes, CAT cell line, and primary lymphocytes, that is PBMCs, show a negligible decrease in the proliferation when compared to the A431 cell lines. Hence, we can say that the CAP has a selective action. Next, we saw the morphological changes, where we see that there is a shriveling of cell membrane and cell detachment, which suggests that there might be a cell death. Next, so there's a hypothesis mechanism that CAP works by the production of ROS. So we wanted to see the total intracellular ROS produced in the cells, and hence we studied the DCFD assay to measure the same, and we see a dose-dependent increase in the ROS production. We validated the same using flow cytometry and can see with the dose scan that there's a dose-dependent increase in the ROS. The time scan showed a transient increase of ROS after 15 minutes post-treatment. Now, to confirm that the ROS is actually acting, we went on to add the ROS inhibitor, NAC, and saw that with the addition of NAC, there's an increase in the survival of CAP-treated cells. Similarly, we studied the same thing with the antioxidant GSH and saw the same results, hence suggesting that ROS inhibition is increasing the survival of CAP-treated cells. Now, ROS can act and damage the DNA. Hence, we wanted to study and quantify the total DNA damage using alkaline comet assay and double-stranded breaks using gamma H2X. What we can see with the alkaline comet assay, that is the total DNA damage, is that there's an increase in total DNA damage at an earlier time point, which corresponds to the increase in the total ROS. With the gamma H2X, we see almost an 8 times increase, almost a 4 times increase in the double-stranded breaks, and we see from both of these that there's a persistent DNA damage even after 120 minutes. And it also shows a slower response, slower repair kinetics. Now, since there's a DNA damage, there should be some sort of DNA damage response signaling as well. So we wanted to study the ATM-ATR pathways. From there, we can see that A431 activates all the four molecules by phosphorylation, that is ATM, ATR, and its downstream kinases CHEK1 and CHEK2. And corroborating the same with the RT-PCR, that is the gene expression, we see that there is no change in the total protein expression, but it is only by the protein phosphorylation that the DDR signaling is happening. So we wanted to quantify the cell death. Now we did the PI staining to see that there's an increase, dose-dependent increase in the cell death, as we see with the subsequent peak. And we can see with the PI annexin staining that is the apoptotic cell death that's happening. So hence it's the apoptotic cell death that CAP is triggering. So this is the total work that I've done, that I've presented here. And so to conclude the whole study, I can say that CAP exposure generates ROS, which causes the DNA damage. And that DNA damage in turn activates the ATM-ATR pathway and the CHEK1, CHEK2 pathway, which can have multiple outcomes. But what we see is the cell death. Further, I'm currently working on the effect of ROS on mitochondria and lipid peroxidation. Role of antioxidants is yet to be studied. Various transcriptional factors regulating redox modulation. And lastly, I want to study the in vivo models as well. I would like to thank everyone from the lab members and all the BRC people over here and everyone over here who gave me the platform to present my work. Thank you.
Question from the audience. Anyone? Okay. Yes, please go ahead. Yes.
Nice presentation. My question is, what is the underlying mechanism for which your cap is behaving differentially in cancer and normal cell? Have
So-
you checked any uptake or anything, how it is behaving differentially?
So what I can think of right now is that it's basically the ROS and antioxidant mechanism. So when it's known that every cell will have some antioxidant levels which can manage the ROS increase, and when we are giving an extracellular, when CAP is giving an extra ROS stress on the cells, the normal cells will be able to manage it in much better way as compared to the cancer cells, where cancer cells are already having an increased ROS.
Okay.
Thank you.
Anyone else? Okay. If there is no question, thank you very much.
Thank you, sir.
Sisukan Suman.
Yeah, I just make the two requests for the speakers of this session that they may like to utilize the pointer which is available on the podium, and also to follow the time they can just see on the screen, presentation screen, so that they can better manage their time.
Good afternoon, everyone, and thank you, Society for Radiation Research, to give me this opportunity for present my research work titled Development of Theranostic Liposomal Radio Nanoformulation for Immunotargeted Combination Therapy of Breast Cancer. For the introduction title, what I have put up is that upgrading the Doxil for clinical use. As we know that Doxil is the first FDA-approved liposomal chemotherapeutic nanoformulation for the treatment of various cancer. And we have upgraded this Doxil by radio labeling with the 99M technetium aspect imaging radionuclide for dosimetry study, and with therapeutic radionuclide 177 lutetium for combination therapy of breast cancer. And further, we have conjugated it with the target moiety trastuzumab antibody and its fragments to target it better and specific to the breast tumor. For the background section, I will quickly discuss how we conceived this project. This lately start from the lately born notion that intervene to eliminate all the cancer cell. Otherwise, intervene tumor microenvironment will lead to metastasis and the resistant development, which lead to the reoccurrence and the mortality of the patient. And the eliminating all cancer cell could be achieved by the combination therapy using the nanoformulation. This is one of the image from one of my review, which shows the different combination radionuclide therapy in combination with the chemotherapy used till now for the treatment of several cancer. Then I asked this question, that can we improve the established frontline cancer therapies at clinics under combination radionuclide therapy regime? Answer is yes, we can do, and this was done by the preparation of target moiety and then conjugating it to the radio level Doxil and further doing the anti-tumor effect. The development of target moieties were done by the enzymatic digestion, pepsin and papain, and radio level. The radio level target moieties were characterized for its affinity to bind to the HER2 receptors. And for the radio labeling of liposomes, what we have done is that we have conjugated the pegylated lipid DSP PEG 2000 amine with the bifunctional chelator CHS DTPA, and the liposomes were found by the ethanol injection method with the remote active loading of the doxorubicin into it. The target moieties were conjugated to the liposomes using the click reaction between the SH group on the TROS modified antibody and the maleimide group on the liposomes, and characterized by the DLS, TAME, PAGE, and multi-mass spectroscopy. and the doxil and the immunodoxils were labeled with the 99 m technetium using the stainless chloride reducing agent at pH 5 and 177 lutetium radionuclide and characterized by DLS TLC. Under the result section, this is the SDS page and this is the HPLC chromatogram indicating the formation of purity of the formation of the fragments. Again, cell binding study, dissociation constant calculation study, and immunoreactive fraction calculation study indicate that the affinity of the target moiety is in nanomolar range to target doxil to the HER2 positive breast cancer. In vivo evaluation, for the first time we are able to show that FAB- and FAB2 of trastuzumab could detect the HER2 positive tumor lesion even at the 3 hour followed by the biodistribution study. And this is published in one of our CBR paper. Further, this is the IR peak showing the different peak for the thiourea bond which is found between the pegylated lipid and the bifunctional collector. Further, this was characterized by the multi-MS analysis which show the successful conjugation based on the difference in the size. And further, this also NMR aspect of 31P indicates the purity of formation of DSP-PEG CHX DTPA. The DLS and TAM analysis of doxil and immunodoxil show the size of different formulation under optimal 100 nanometer and the zeta potential of approx -30 indicating the stability of the formulation. This is the SDS space showing the successful conjugation based on the size. And this is the HPLC chromatogram showing the successful conjugation based on the retention time in the column. Again, this is a multi-MS spectra showing the successful conjugation between the target moieties and the liposomes. Once the doxils were characterized, we want to show that our immunodoxils were only specific to the HER2 positive cells. We have shown this by confocal microscopy in which we can see that our immunodoxil is showing more uptake which get reduced in presence of antibody, receptors are blocked. Another beautiful experiment what we have done is that we have grown the HER2 positive cells and HER2 negative cells together and we were able to show that our immunodoxil only go into these round shaped cells which is HER2 positive and not in this spindle shaped cells which is HER2 negative. So this proves the specificity of our nanoformulation. This is again proved with the immunodoxil 5-2 and the same as condition is also done with the cell binding study to show this specificity of our immunodoxils. In the in vivo evolution, we are able to show that our immunodoxils show more tumor uptake and faster pharmacokinetics as compared to the doxil, and FAB2, thus two immunodoxils show the faster pharmacokinetics and better tumor penetration as compared to the antibody doxils. This is published in one of our paper in the DDR journal. Once the specificity of immunodoxil was confirmed, we have also shown that lutetium and doxorubicin shows enhanced combinatorial therapeutic dose at lower doses by viability assay, and in cell cycle analysis, mostly G1 arrays was found at the different doses of doxorubicin and lutetium using the concurrent therapy in the liposomes. We have also shown that immunodoxils cause the HER2 specific toxicity in 3D spheroid culture and the 2D cell culture. As you can see in this graph that immunodoxils show the maximum cytotoxicity in the SKBR3 and SCOP3 which is a HER2 positive cells, and it gets reduced in the presence of excess antibody because it is blocking the receptor. So this doxil, immunodoxils are especially causing the specificity which is HER2 specific. In the tumor regression study, lutetium lipdox is showing the minimum tumor regression as compared to lip5-2 dox is showing the maximum, which has also the better tumor penetration and better pharmacokinetics. In the conclusion slide, 4 points I have put up. The first that our target moieties FAB- and FAB2 can detect the HER2 positive tumor lesion even at 3 hours, which is very good for the clinical setup. Patient can go on the same day after doing the dosimetry study. Doxil, which is used in the clinics regularly, is labeled with a technetium so that patient can undergo dosimetry study before they can go for the treatment. Then doxil conjugated with trastuzumab so enhance HER2 specific tumor uptake and cytotoxicity, which is also HER2 specific compared to doxil, and FAB2 conjugated immunodoxil so better tumor uptake and the faster pharmacokinetics and cytotoxicity as compared to the antibody conjugated doxil. These are my acknowledgements, and thank you to my head RPS, the BRC, and the head RPS, our section, and Chandan sir for their help in the entire study, and finally Society for Radiation Research, again to give me this opportunity and a platform to show my work here. Thank you everyone.
Thank you. Question from the audience, please.
So basically what we are formulating the doxyl conjugated nano formulation. So how we are checking that the doxyl is not getting released when you give the treatment to the cells? Since what happens that if doxyl release, the kinetics of uptake is going to be different than when they are conjugated.
For that, there are two criteria what we have maintained. First, we have checked the doxorubicin release in the serum condition in vitro. When in vivo, when we are doing the imaging study, we are extracting the tumor and we are extracting the doxorubicin from it to show that our radionuclide and doxorubicin is correlated with the amount, so that the doxyl is not ruptured and our radionuclide and doxorubicin both together is reaching the tumor. This is a control for the in vivo study. For in vitro study, we are doing the radio level serum stability at up to the 144 hour for the 177 lutetium radionuclide and up to 24 hour for the 99M technetium because of their different half-life.
But in vitro conditions, whether you are testing, since it may happen that we treat with in vitro, doxorubicin may be released from the, and then uptake is going to be different. In that case, if you do this sort of either studies where you have got seen that doxyl as well as your radiopharmaceutical are together, they are in the cells.
We have done the drug release study in the serum plasma condition.
Not serum, in culture medium I am telling. So in vitro conditions, if you are treating, if doxyl get released from the nanoformulation, uptake is going to be very faster.
Yes, sir.
So that part you can take care if you have not done it.
Yes, sir. Thank you, sir.
Thank you, sister. Okay, go ahead, please. Brief question, please.
This part that doxorubicin is a tomosomragitiva, this is a very known fact. So we are not focused on that part. So we have not gone on the molecular basis. We are seeing the tumor regression and the in vitro toxicity, and we are much more focused on the targeting the doxy to the tumor and in the combination with our therapeutic radionuclide. I agree with that. Being a doxorubicin is a very established drug, and even doxil is a very established drug in the clinics. So we have not gone much into the molecular part.
Okay, thank you very much.
Thank you everyone.
Next
Right.
candidate is Venkatesh Babu, and most are, okay, he's here. Come in, please. Amit, I'm not having his document, so you have to give me later.
Good afternoon to one and another person here. My name is Venkatesh PS. I am here to present the topic entitled Bacterial Supplementation as a Strategy to Mitigate Pelvic Radiation-Induced Neurotoxicity, a Comprehensive Preclinical Study. So, radiation exposure can happens into different situations such as accidental public exposure, occupational exposure, and medical exposure in the terms of radiotherapy. So, as we all know that about 50 to 60% of the cancer patients receives radiotherapy as part of their treatment strategy. And with respect to the pelvic radiation, which comes under the pelvic region such as gynecological, urological, and gastrointestinal tract, which leads to types of side effects which leads from the acute to chronic stages, which is mentioned here. And which further leads to the decrease of the quality of the life, which includes the financial challenges, hospitalization, emotional distress. So when the radiation administered into the gastrointestinal tract, initially it will start with the nuclear DNA damage, which further leads to the apoptosis, and which leads from the acute to the chronic tissue damages, includes mucosal barrier breakdown and fibrosis. And further it leads to the disruption of the microenvironment in the gut, which leads to the dysbiosis. So as we all know that gut microbiota, which resides in the gastrointestinal tract, which is a collection of bacteria, archaea, and various other microbiota, which helps in the immunomodulation, integrity of the gut barrier, and interference with pathogens, which all helps in all these particular aspects. So further, there are many studies which says that the radiation can changes the gut microbiota, as well as there are the lot of studies which happened wherein the good bacteria such as lactobacillus, bifidobacterium, and streptococcus, which has the potential in maintaining the hemostasis of human. So further, it is very interesting that research have been developed into the in finding that the relationship between the gut and the brain, which is called as the microbiota gut-brain axis, which communicates from the gut to the brain with a lot of mechanisms. And when the radiation happens, there could be a disruption of this gut and brain axis. And further, we are interested to look into the bacterial supplements, which is the live microorganism which can take in adequate amounts, which impose a health benefit to the host. So when we look into the literature, it has been studied that this kind of bacteria has been served as for the integrity and the betterment of the human gastrointestinal tract. Within this, we hypothesize that when the pelvic irradiation happens, is there any gut dysbiosis, or this gut dysbiosis or the microbial metabolites produced by this gut dysbiosis leads to neuronal damage, and whether this can be mitigated through the bacterial supplementation. To aim this, first we have tried supplementing a commercial bacterial composition to the C57BL6 mice, wherein we have found that the radiation has been damaged the gastrointestinal tract, as well as the combination of the bacterial supplements mitigated. Further, we have checked whether the radiation has any damage in the neuronal, and whether the supplementation has any protective effect. And hence we found that the radiation has increased the pyknotic cells in the different regions of the brain, and the bacterial supplementation has the protective effect on it. And further, we have looked into the neuronal inflammation as well. We have found the same thing, and finally we have come to the end that this bacterial supplement has the protective effect in the radiation, whole body radiation induced intestinal damage, neuronal damage, and neuronal inflammation, which has been communicated in clinical and translational oncology. And we want to find out what is the involvement in the gut microbiota and with respect to the pelvic region. So further, we have made a strategy wherein we have exposed the animals only respect to the pelvic region, which is shown in the figure. And we have further looked into the behavioral as well as the gene expression of the brain. And we have found that yes, the bacterial shift has happened after the radiation, which has a lot of link to the brain as well. And finally, we have found that the pelvic radiation has damaged the neuronal as well in the terms of neuronal survival, neuroinflammation, and neuronal maturation. And again, we have looked into the behavioral changes, wherein we have found that the rats which we have exposed to the radiation has more close to the darker side of this setup, wherein we have found that the exploration is very less. Also, we have found that there is a loss of NMDA2, which is responsible for the neuronal plasticity. And this we have reported in the journal Chemicobiological Interaction. And finally, we want to know whether this bacterial supplementation can mitigate the pelvic irradiation induced neurotoxicity. For that, we have created another animal model where we have implemented these bacterial supplements, and we have found that there is a mitigation in the bacterial gut microbiota, as well as we have found that there are a lot of shift in gut microbiota happening in the radiation induced as well as the supplementation group. Further, we want to look into that how this bacteria, the gut dysbiosis leading to the neuronal toxicity, wherein we have looked into the leaky gut with respect to the parameters such as the integrity as well as the different tight junction proteins such as occludin and zonooccludin, wherein we have found that the radiation has induced the leaky gut, which is mitigated by the bacterial supplements. And further, we have looked into the gut inflammation factors such as IL-6 and TNF-alpha, wherein we have concluded that these inflammatory factors also can be a factor, one of the factor for the neuroinflammation, and this work has been submitted to the International Journal of Radiation Oncology, Biology, Physics. And further, we have looked into the histology and the behavior as well as wherein we have found that this bacterial supplementation has the ameliorative effect. And as I told, the bacterial supplements, this has the metabolites which are produced by the gut microbiota, wherein we have found that certain pathways which has altered and which has mitigated after the bacterial supplementation, and the major bacterial metabolite groups such as long chain fatty acids, then aryl thioethers, and so many others which has been having a protective effect after the radiation treatment. So finally, to conclude that, in the pelvic radiation strategy, we have found that there is a loss of intestinal integrity, and which is creating the leaky gut, which releases all the inflammation-inducing factors to the brain, and which has been ameliorated after the bacterial supplementation in all the aspects of brain metabolomics as well as the gene expression we have validated. So to conclude, we can say that the bacterial supplement can be one of the treatment strategy for the pelvic radiation-induced neurotoxicity, and this is our few research work published and key references. I would like to thank our director, Dr. B.S. Satish Rao sir, who has given critical comments on this work, and also my supervisors, Dr. Kamlesh T. Mumbraikar and Dr. T.S. Murali, then all the clinical collaborators and my colleagues, then especially ICR at 2020 for the opportunity to present my work. Thank you.
Question from the audience, please.
But which has been reduced after the bacterial supplementation treatment. Yes, yes, ma'am. Yes, ma'am. Yes, yes. Particularly long chain fatty acids. Yes, yes, ma'am.
Any other?
How do you correlate behavior with histology of the brain? I mean, how do you do that? Behavior is so complex.
Yes, sir. So we have looked into the different stages. So first we have seen, is there any connection which can happen from the gut to the brain? So we have found that yes, there is a leaky gut. Then we came to the tissue level as well. So we found that there is a neuronal damage as well as the inflammation happened. Then finally we checked the behavior. Is there any change in the behavior? But not respect to all the factors which we have found in one factor, wherein we have found that the exploration is very less. So we are suspecting that because of the pelvic radiation induced damages created the less exploration. Thanks.
If there is no question, thank you very much.
Thank you, sir.
And next candidate is Preetam Samal.
afternoon, everyone. Thank you, Society of Radiation Research, for allowing me to present the topic on the in vitro modulation of radiation-induced DNA damage and repair by TLR agonist mannan oligosaccharide. So, talking about the TLR, what are TLR? TLR are toll-like receptors mediated from the pattern recognition receptors that are found in various immune and non-immune cells. So, brief pathway is given in the right side corner. So mediated independent pathway and dependent pathway follows the TLR signaling. Talking about mannan oligosaccharide. MOS is a TLR agonist, which is a carbohydrate derived from cell wall of yeast, and it improves the gut health, enhances the nutrient absorption and response as a prebiotic. Talking about the structure of the yeast cell wall, having chitin layer, beta glucan layer, mannan protein and mannan in the network-like structure. So what is the role of MOS in changes of the gut microflora? So talking about the MOS, it helps the beneficial microbiota such like lactobacillus, E. coli, salmonella. So in this goblet cell increases, villi height also increases, that helps in decreasing the crypt depth. So the mucin protection also increases. In this case, that it inhibits the pathogen microbiota colonization. So and also it increases the interleukins, so it boosts the immune responses. Now talking about the effects of radiation on the cells, biological effects. So we are talking about mitochondria and the nuclear DNA. So in mitochondria, what radiation does? It hampers the electron transport chain, which eventually hampers the ATP-ADP production and have a great effect on the mitochondrial dynamics. And in the nuclear DNA, it hampers the single and double strand breaks, and DNA repair can also occur by NHEJ and HR pathway. If DNA repair is not possible, then cells go to apoptosis. Now talking about the methodology, I have worked for different proteins of DNA damage and repair and also oxidative stress by flow cytometry and different staining and bacterial culture. Talking about the bacterial culture, we have in the left hand side, we have the motility of the bacteria by stab culture. So different motile and non-motile bacteria are present, as we can see in the left side corner picture. And in the right side, I have the fecal metal staining of mice. So negative staining and gram staining of fecal metal staining of mice are present. And the right upper side corner is the negative staining, and down is the gram staining. Now in the bacterial culture, in the different irradiated mice, I have chosen to work. So first picture is the first right side corner is the in the left corner. So radiation alone, drug plus radiation, and treatment and drug alone treatment were chosen. So we can see the difference in the size of the number of colonies and the patterns of the bacteria's shape and sizes of the bacteria. In the downside, I have the prebiotic effect of MOS. In the left side, we have a well division method of choosing the beneficial mycobacteria. So how MOS benefits the beneficial mycobacteria like Bacillus megaterium and Escherichia coli, E. coli. In the right side corner, the antagonistic effect of MOS to inhibit the pathogenic mycobacteria like S. aureus and Corynebacterium glutamicum, as zone of inhibition is formed by different concentration of MOS. So talking about the changes in the cell lines, NKE cell line, normal kidney epithelial cell lines. So in the left side, we have the morphological changes of NKE cells after 24 hours of different treatments like control, mannan, 3 gray, mannan plus 3 gray, 5 gray, and mannan plus 5 gray. In the right side, we have the changes in the DNA damage and repair proteins like phospho-check1, gamma H2X I have taken for 24 and 48 hours, cleaved PAR for apoptosis pCNA, and loading control. So talking about the cell cycle study, we have cell cycle study for various treatments as mentioned before. As we can see, mannan plus 5 gray, mannan plus 3 gray, both in 24 and 48 hours, having G2M arrest, so cell is preparing for the repair. Talking about the levels of DNA damage by alkaline comet assay, I have assayed for 3 gray and MOS plus 3 gray. As we can see in the picture, MOS plus 3 gray has less comet, so less damage is formed. And in the right side, I have depicted the Ross generation by CMS2 DCFD assay. Also in the 3 gray and MOS plus 3 gray, we can see a significant difference in that. Now summarizing, so the bacterial colonies moss demonstrate the prebiotic potential by supporting the gut microbiota. Also by different staining techniques, it have shown the effectiveness, and also in the cell lines, moss protects the NK cells from the radiation-induced damage, DNA damage, and oxidative stress. So I would like to acknowledge my director, Suri Channa sir in Mass DRDO, director in Mass DRDO, for providing me the facility to work, and thanking DRDO for funding and providing me the fellowship. Thank you.
Thanks. Question from the audience, please. Anyone? Okay. If there is no question, I would like to thank you. Thanks.
Thank you, sir.
Rajhans Kumar.
Good afternoon to all of you. So I'm Rajesh Kumar, working as a medical priest at Aiims Patna. So my title is Comparative Dosimetric Analysis of Two Volumetric Modulated Arc Therapy Techniques for Craniosacral Irradiation. So since when 2011 BMAT has described from that time, so and most of the center out of 12, 8 centers are using larger arc length for the spinal cord. So mostly 200 to 360 degree they are using. So actually the craniosacral irradiation, the planning is very challenging because of the lengthy target volume. So BMAT techniques include full arc and combined arc. In this, I have evaluated the dosimetric benefit of the volumetric modulated arc therapy, the combined arc over the full arc. So BMAT offers superior dose conformity and it does not require the junction between brain spine and spine, and it will also minimize the organ at risk dose. So CSI also, it presents challenges due to extended target volume, and in this also we evaluate the dosimetric benefit and combine arc BMAT techniques over the full arc, optimizing treatment efficacy and dosimetric. So BMAT techniques offer superior dose conformity index, and also it requires not junction between brain spine and spine compared to 3D CRT. So in objective, we evaluated the CSI, BMAT CSI for the two approaches, that full arc and combined arc. Combined arc is nothing but it is that full arc for brain, and that you can see that non-covalent vertex arc for brain. This is half arc. As I use half arc, you can see. Sorry. Yeah. So and also first time I reported that body type arc. You can see here back side that 20 degree avoidance. First time I have reported. First 20 degree avoidance in spine region. So it will reduce the OAS dose, and 50 to 90 degree AP arc I have used. First center I kept in this one area, cervical spine area, and second ISO center we will keep according to the PTB length. If PTB is more than 60 centimeter, then second ISO center I have to put for that, and otherwise first ISO center for spine, first ISO center for the brain, it is okay. So CT simulation has done for 10% on G Optima, which is 16 slice thickness, 3 mm slice thickness has taken for this patient, and thermoplastic mask for immobilization we use, and for OAR's contour and PTB, so RTOG guidelines and Atlas guidelines, ACNS 0331, the clinicians is used, and 2 BMAT created here with the 6 MB energy and by the single medical physicist. Full arc and combined arc, which is non-covalent vertex arc and body type arc. So the prescription dose was the 36 gray in 20 fraction, 1.8 gray per fraction, followed by boost to tumor bed, 23.8 gray in 1.8 gray per fraction. So parameter evaluate 95% of isodose and B107, the volume received by 107%, and conformity index, which is the conformity index is that 95% isodose line divided by the PTV volume, and homogeneity index the dose received by 2% of PTV volume minus dose received by 98% volume divided by dose received by 98% PTB volume. And organ at least like eye, lens, optic nerves, thyroid, heart, lungs, liver, kidney, ball, parotid, esophagus, etc. So as you can see that this is in first figure, this is the isodose 50% isodose line of 36 gray of full arc, and the second figure is the isodose line of the 50% isodose line of the body type arc. So in second figure you will see that how beautifully covered the PTB volume, and it is able to the spare OS dose as well as. So I did the correlation between the full arc and combined arc of all those ORs. As you can see that the right eye for right eye and left eye, that combined arc, it is statistically significant. It is reducing 35.1 to 32.85, 34 to 32.6, and lens dose also it is statistically significant in left 8.78 to 8.1, and right lens also it is reducing 9.5 to 8.2. So if you say in thyroid, heart, lung, liver, so it is not statistically significant, but it is reducing in combined eye, it is reducing. So, but if you will see the B95, it is superior in full arc if you will compare to partial arc. But in case of B107, so it is slightly reducing in full arc compared to partial arc. But in case of the conformity index, conformity index is slightly good in full arc compared to combined arc, and homogeneity index is better in combined arc if we will compare to full arc. And homogeneity index, if you will see the homogeneity index, it is slightly good in combined arc. MU is more in that partial arc compared to the full arc, but it is not statistically significant. So in this conclusion, the combined VMAT plans utilizing a non-coplanar vertex arc for the brain and body style partial arc for the spine demonstrated significant reduction in radiation dose to critical organs ORs, while maintaining the PT coverage, dose homogeneity and conformity index comparable to the full arc plans. Although the combined arc plans require slightly higher monitor units than the full arc plans, they offer better ORs sparing, making them a more efficient option for minimizing toxicity in CSI. So the author expressed profound gratitude to all the patients of this study and the Department of Radiation Oncology at Patna, without whom this study was not possible. Thank you. Any questions?
Thanks. Question from the audience, please.
Ma'am, I can. Sorry, ma'am.
Nice presentation. So did you compare the integral dose between the two plans?
Integral dose I didn't compare, ma'am. So,
But you should
I am sure. But in partial arc, that I have seen that if you will see Jack Jens study and Biplav et al. Study, integral dose will be less in this. They used half arc, but I used partial arc. But I am sure 100%. So integral dose will be less in partial arc compared to full arc.
Yeah, because when you are doing a magnafield radiotherapy, integral dose to the body is very important.
Yeah, if integral dose, what this will be this part of research. You have to crop from the PTB that part, and you have to within 10 gray you will get.
Okay. And second question, during the dry run for both the plants, were there any difficulties? Because at times you have problem with the couch and all that. So
couch, ma'am, only couch angulation I used only in brain only 90 degree. So from console you can do 90 degree. Otherwise I used clockwise, anti-clockwise direction. Only Gantry will move, the radiation will deliver in that junction. Radiation will not deliver. Again it will move like that.
Okay, thank you.
Couch angulation was not there, but only for brain it was.
Any other question? If there is no question, thank you very much.
Thank you.
So last candidate is SN Behra.
A very good evening to all of you, respected chairperson, dignitaries. I am Sachidanand Behra, scientist working at SS Hospital and Research Institute, going to present my topic, chronic sunlight UV radiation as a catalyst in the pathogenesis of post-Kala-Azar dermal leishmaniasis among VL endemic population of Bihar. This work was done during my PhD, and it was done at ICMR RMRI Patna with field and lab setup with support from medical officers and field support staff with some help from Dr. J.K. Singh Group. Coming to the disease, you can see here that leishmaniasis is a parasitic disease but is very much endemic in the Bihar region, and now the other form of this disease, not VL, it is now more PKDL. VL is the visceral leishmaniasis. You can see that we are having pointer. We are having VL with organs like liver and spleen affected, but this disease is little bit reduced. Now PKDL, the skin lesions are now very much prominent. This disease is being carried by the vector sandfly, and the main problem is leishmaniasis elimination program. It is being threatened by the PKDL, and it is the main challenge by the government of India to control leishmaniasis disease in India. This is the region. This is the region where the endemicity of Kala-Azar or other form that is PKDL is prominent in Bihar. Still, we are not able to eradicate this disease, although we have controlled the VL number of cases. You can see what the connection between VL and PKDL. The main thing after treatment of VL, the patient get converted to PKDL, the dermal lesions, and there are many other factors. One reason may be the exposure to some sunlight, and there is some hypothesis earlier. That is why there was a program by the ICMR and the DST to see the health and climate related aspect correlation. So we conceived this thing from the increasing cases of PKDL, and now in India the PKDL cases are more in Bihar only, and neighboring areas where migration is taking place, this disease is also getting spreaded. So UV radiation all knows, but how it is connected to PKDL? The PKDL cases, they are more or less belonging to the social low classes people, and they work in the means labor and field worker type, and that is why they are getting highly exposed to the UV radiation of the sun. And this observation of 2-3 years preliminary data gave us the inspiration to see, is there any connection of PKDL development with the sunlight? So this factor was supported by some earlier hypothesis not done anywhere else in India, because the endemicity is here in the Bihar, and RMRI is the WHO referral center to investigate leishmaniasis related research and disease. So we have a research literature where there is effect of UVA and UVB on the skin, and we can see in the mouse UVB is responsible for carcinogenesis. But if you see, when there is much of UV radiation is coming, the effector pathway is getting blocked and suppressor pathway is getting activated. So UV induced systemic suppression is taking place, and it is known for cancer and other disease, but it is not known for infectious diseases like leishmaniasis or any other disease. So I started this thing in 2016, where we have taken two methods of calculating the UV radiation from the sun naturally by the help of UV light meter, and also with satellite based method with the help of the IMA and the KNMI team. We have validated this thing with manual measurement, and we got the approval from the ethics committee and other government of Bihar and other regulatory bodies. We have covered almost 4 districts, including Vaishali, the highest endemic one, and we have screened 358 patients, and finally we could include 283 patients. We can see the measurement of this UV radiation varies in different season, but the season is highest in the pre-monsoon time, particularly in May area, May and April, and it is less in the winter time. With every single day we have measurement at different locations at the study sites, and we are having a big team with this, and I was a leader in that area relating to data calculation, patient visit, and other relating coordination to this particular scientific validatory work. you can see in the right hand side, there is a similar trend of UV changes in the seasons, and we see there is a climate change, particularly sun radiation coming high. Now it is coming to the exposure to the population. We can see in Indian region, it is highest around the June time, the pre-monsoon time in Bihar. Bihar expect the monsoon around August and September. So at that time it was highest, you can see here, and it gets gradually down. We got the overprice file personally on the request from the KNMI. I have a collaboration with them, and I got the daily recording as well. So from the questionnaire based data, I can find there are the cases, the patient particularly, where labor, field worker, those who are getting more exposed to the sunlight. And if you see on the right hand side, you will see particularly the sun sensitivity is high. 207% got sun sensitivity in comparison to 76. This is the sub-classification where we get the clinical form that is more macular, but 24% nodular polymorphic cases. We see the cytokines, and it was a good, means elevated in the picadial cases in the comparison to the healthy one. And we have profiled the vitamin D and iron. They are helping in the immunoresponse pattern for getting healed, and we see a significant change. Apart from this, we checked the cell lines, and we have experimentally given the radiation 5 minutes and 10 minutes. The 5 minutes was LD51, and for 3 days it was highest. And we have found that the expression of THP1 was like in every 5-minute radiation, the cells are having TLR signaling and IL regulations very much significant. If you come to the subsequent passage, we find that after 3-4 passes under stress to a particular group of UV radiations, there is a significant change, particularly in IL-10 and IL-12, that indicates UV radiation does a change. And we have infection data, but that slide is not given here. This is the facts data where we have seen TL2 and TLR4 expression. In the upper panel, here is the healthy one, and here is the patient one. And these two, this is low radiation, this is high radiation, this is low radiation and high radiation. Low radiation means it is less than 2 hours, high radiation means it's more than 4 hours. You see, TLR2 is 2-fold change, and if you see the TLR4, this is also 1.5-fold in comparison to quantitative data is here. This is the quantitative data. Here we have also seen the same. Sir, 30 seconds. We have checked the immunoglobulins, and this is the hypothesis that is saying that these regulations ultimately lead into some sort of immunosuppression. And we have checked some SNPs also. This is published, and this is the summary as you have seen. And this is the highlight. Our policy is adopted by the Government of India for protection purpose as a prophylaxis. And I will acknowledge my collaborators, Dr. J.K. sir, Dr. Krishna Pandey, and my guide, Dr. Wahabali. I have few awards, and I have published 2 papers, another origin group. I have total 12 papers, and I'll thank my group for this. And this is my team for the whole entire field work. Thank you so much.
Question from the audience, please. Any? Okay, if there is no question, I would like to thank you.
Thank you so much, sir.
Before closing the session, I will clarify to all candidates that audience number was small, and for few candidates there was no question from the audience. So I will discuss with other members, and we will find out one reasonable solution that if in case audience did not ask, candidates should not be penalized. So we will make appropriate compensation. And with that, I would like to thank all candidates and close the session. Thank you very much to the audience.
Thank you, sir, and thank you all the participants. Next, we move on to the poster session. I would like to call upon stage Dr. Anita Kumari to chair the session, and poster presenters from number 1 to 15 will be presenting in this session. Each person will be allotted 4 minutes, and the timer will be visible in the screen. And due to time constraints, we would like to restrain 1 questions per presenter. First presenter, please come up on stage to upload your poster, and the rest, please approach the person near the podium to upload your posters. Thank you.
Thank you, Dr. Jani. Before wasting time, move to our poster session, and for this there is a 5 minutes, 4 minutes for presentation and 1 minute for question answer. And our first candidate is Dipali Patel.
Good afternoon, everyone. Thanks to organizer give the opportunity to present my research work. My topic is optimizing heart and lung sparing in synchronizing bilateral breast cancer radiotherapy, a dosimetric and radiobiological analysis of 3 different fractionation schedules. And my study object, there are two study objective. Primary objective to compare the dosimetric parameters and normal tissue complication across 3 fractionation in synchronizing bilateral breast patients. Secondary objective is my study is evaluate the heart and lung toxicity risks through the NTCP modeling, analyze dose volume relationship using parabolic equation, comparing PTB coverage and organatrix doses. Methods and material, we are retrospectively selected 5 synchronizing bilateral breast cancer patient. Study period is 2021 to 2024, and this daily CBCT verification done in all patients. Treatment planning, we are using the Eclipse treatment planning system to creation of the plan. 5 VMAT partial arc VMAT plan with the dual isocenters we are using, and 3 different fractionation schedules. Conventional fractionation dose is 50 gray in 25 fractions. Hypofractionation dose is 40 gray in 15 fraction, and ultra hypofractionation dose is 26 gray in 5 fraction. And dosimetric plan evaluation parameter, we have done that in terms of the target coverage, homogeneity index and conformity index. And organatrix assess the heart mean dose less than or equal to 5 gray, lung mean dose less than or equal to 15 gray, and V5 less than or equal to 65%, and V20 less than or equal to 30 gray. LAD mean dose is less than or equal to 25 gray. The analysis, biological analysis done in NTCP modeling, we are using 2 models, Poisson LQ model and Lemon Culture Berman model. Statistical analysis done in crucial wall test, and these fractionation, these 3 fractionation correlate with the mathematical parabolic equations we are using. And table 1 represents the patient characteristics we are involving, and table 2 the PTV dosimetric parameters, and table 2 is organatrix parameters, and results showing the NTPC value in table 3. These are our isodose line distribution in 3 different fractionation schedules, and lower is the dose volume histogram. And the results in our in terms of target coverage, significant decreases in V105cc from conformal fractionation, conventional fractionation to ultra hypofractionation, improve homogeneity index with the ultra hypofractionation, and consistent coverage across all fractionation schedules. Organatrix protection, significant reduction in lung doses with ultra hypofractionation. V5, V10, V20 shows consistent decreases, and mean lung doses significantly lower in ultra hypofractionation. Heart dose also decreases in V5, conventional fractionation to ultra hypofractionation. And radiobiological outcome NTCP analysis, the heart mortality risks reduced in 3.5 times from conventional fractionation to ultra hypofractionation, and pneumonitis risk also decreases in 2.7 times from conventional fractionation to ultra hypofractionation. And significant reduction in symptomatic pneumonitis within the 6-month, long-term fibrosis risks and combining lung complications. And conclude my research work, ultra hypofractionation demonstrate the superior dose uniformity and organometrics protection and similar biological effectiveness across the three different fractionation scheme, notable reduction in cardiac and pulmonary complication probability, clinical validation through extended follow-up needed, and findings suggest potential benefit to ultra hypofractionation in simultaneous synchronized bilateral breast cancer radiotherapy. And then coming to limitations, the small sample size, radiobiological modeling limitations, model parameter dependencies, and interpretations causing comparative metrics only, not definitive clinical outcome predictor, individual patient response variation possible.
One question allowed. Yes, you come forward.
It's good as a work and comparing the three models are very good. Given this result, what do you want to practice from tomorrow onwards?
Sir, in our hospital, conventional fractionation we are also using, and hypofractionator we are also using. And unilateral cases, we are recently start ultra hypofractionation schedule.
No,
26 gray in 5 fraction.
it clearly shows there's an advantage, and also there are many literatures which are really showing. There are many literature which are showing there's a definite advantage. Would you like to start using it in a routine work in your hospital? Unilateral also would like to.
Only one question is allowed. Now moving to our
Thank you.
second participants, R.S. Karki. Yes, you go ahead.
Good afternoon. I am warm greetings to all the participants. Namaskar. I am from Nepal. I am Ram Saran Karki from Tribhuvan University. Currently, I am working as a researcher in Nepal Academy of Science and Technology. I am especially working in this thermoluminance dosimeter. We are using thermoluminance, this TLD reader 6600 plus, and I work to calculate uncertainty in this TLD dosimeter, and I use this TLD, and we expose our TLD in this LINAC system, variant LINAC system, which is present at Bhaktapur Cancer Hospital. About the uncertainty of this TLD, both batch and reader, we first develop measurement model, and we perform about 9 type tests. Among them, this energy and angular dependence, fading, and non-linear response is the major contributor for causing uncertainty. And about this energy and angular dependence, we use different angles, and we expose our TLD in cobalt 60, X-ray with different energy, and cesium 137 source, and found that this energy and angular dependence is the major contributor. And we found, first of all, we developed measurement model, and we don't have SSDL in our country, so we expose in overseas our TLD card, so we subtract background for each TLD to calculate uncertainty, and found that due to expose in this X-ray in airport, this background also contribute for uncertainty, and overall uncertainty for k is equals to 1, we took for k is equals to 1, it is found to be 19%. And we compare with the guidelines which is provided by IAEA, that is less than this is within the range which is 1.5 factor which is proposed by IAEA, and we use this TLD to calculate uncertainty in Bhaktapur Cancer Hospital. We expose our TLD in two different energies, 6 of photon of 6 MB and 15 MB, and we placed we studied on the basis of field size, and we took 4 field size, 10 by 10, 15 by 15, 20 by 20 and 25 by 25 field size, and we expose our TLD in two different energy in 1 centigrade dose, and we placed 3 TLD inside the field size and 2 TLD outside the field size, and which is 5 centimeter from the edge of the field size. Even we found some recordable dose outside the field size, and we found that larger the field size, dose received by TLD is more, and the uncertainty is within the 5% range inside the field size, and this I want to conclude that this
time is over.
Okay.
You completed.
And this highlights the influence of field size, energy level and potential variation on relative SP10 dose measurement. This SP10 we normalized with respect to exposed dose, and larger field size and higher energy else uniform dose distribution significantly reducing uncertainty at the central position. Okay, thank you.
No question is allowed. Okay, thank you, Dr. Kartikeya. Our next presenter is Dr. S.H. Dhobi. Next is Dr. Biswajit Das.
A very good afternoon to all of you present here. My name is Biswajit Das. Okay. Okay. I am working as an assistant professor in the Department of Physics, Vidyas University of West Mendipur district, West Bengal in India. Now I am going to deliver a review study on the relation between the radon exposure and basal cell carcinoma skin disease in human body. It is very well known to us, to all of us, that basal cell carcinoma is a type of skin cancer. It begins in the basal cells beneath the epidermis of our skin. Most frequently, it is appeared in a portion of our body which are exposed to directly sunlight, and few parts of our body will also be exposed or develop BCC which are not exposed to sunlight, for example genitals. Actually, like many other malignant diseases, BCC also develop due to the mutation in the cells, mutations in the cells, so that the DNA of the cells will be damaged. The main risk factor is the ultraviolet radiation exposure, and less risk factor is exposure to arsenic, tar, coal, paraffin, certain type of industrial oil, radiation and radiation therapy, and also immune system function decrease. However, exact cause for developing the BCC may not be clear in some cases. Though not only basal cells are present in the human skin, but also it presents in our lungs. And the International Agency for Radiation on Cancer, IRC, in 1988 determined that radon exposure is directly related to the developing the lung cancer. So this is not the study. Many other studies develop the lung cancer and the radon exposure are directly related, and they have been proven the cases. So actually, lung cancer developed due to the collision between the alpha particles with the cell in the lungs. As basal cells are present in the skin, in our skin, so I think human skin also be exposed with the radon, environmental radon exposure. Here a study also developed the radiation dose level. Here is given here, Etawa Hansa in 1991, and many others. Research study also show the how many doses received by the skin in the level of the 20 becquerel per meter cube in indoor or environmental radon exposure. For this, we have I have reviewed some studies which are published up to 2024. We have some studies. Most of the studies are reported the positive and significant correlation between the BCC developing and the radon exposure. Few studies also reported the positive relation between the basal cell carcinoma and the radon exposure. Few other studies also do not agree with this comment, but many studies also also shown that the relation between the BCC and the radon exposure. So this is my research. I have concluded some summary. First of all, radon exposure is a possible risk factor for inducing several types of skin cancer. Evidence for radon-related skin cancers among minors are scanty. Environmental, ecological, and other studies reported association of high level of radon exposure and BCC. More studies are required. Actually, epidemiology studies are required for confirm the relation between the BCC and the radon exposure. Thank you.
No question is allowed. Now moving to our next presenter. You please come one by one in order to save the time. Ankit Srivastava, Ambika Sandalia, Sivani Behra. You please come one by one. Sivani Behra, Vishal Chabra, Devidas Lehmaga, Vidyapati Jha, Kunti Tandi, Sikhashri,
Good evening, everybody. I am the Chief Assistant, Bagpur Cancer Hospital, Kathmandu, Nepal. I am Vidyapati Jha. So my research, this is for the breast cancer, and I did it in 2020, perhaps 2021, during my PhD work for the hybrid IMRT. So I have taken 40 breast cancer patients, 20 left breast, 20 right breast. This paper is already published in ABCR, Advanced Breast Cancer Research. So in details, you can see here is the site. Next. And so my entire I have done field-in-field technique. I have applied because we have problem. We have 100. We are treating 120% per day in the single linac. Yes. So we have problem there, and we tried at that time whether this technique is applicable or this is useful for the patient or not. And finally,
20%, and I have used one hypofractionation and other is the conventional fractionation, 50-25 and 267 in 15 fraction. So this is the volume. For there are three category of patient. The breast size, the chest wall is different because most of the patient they comes in our hospital after mastectomy, means advanced stage. So the chest wall is treated here. So this is the histogram, but I have shown over there, and this is the sagittal and all three phase images. So this is in detail. So the PTV, what is the dose, and I have measured main 2 points, the CI and HI. So the conformative index and the homogeneity index. So these 2 parameters confirm whether what type of planning we are doing. And the quality of planning is just justified. This is the tool for the justification of the quality of planning. And I found the 99 more than just our main target is the B50, B95 means 95% dose should go in 95% volume of the PTV, planning target volume. So we found in most of the patients about 99% dose is reaching in at least above 95% volume of the PTV. And we found in right breast that planning is the better in comparison to the left breast, because in left breast we have always just more tension about the hard dose. And this is the volume that I have already mentioned. So conclusion, the present study confirmed that the use of field-in-field radiotherapy technique for breast treatment leads to the homogeneous dose distribution in the planning target volume. So the FIF technique provides dosimetric advantage and requires less planning time in comparison to IMRT and others, because we have to maintain the patient load. Additionally, the implementation of this technique in the radiotherapy department is straightforward, resulting in overall dropping dose to the OR, organ at risk. So however, our study has some limitation, including some small sample size and the lack of assessment of the clinical parameters, especially for the OR complications. Now this is the acknowledgement. Thanks.
Presentation is nice, but this is not in a poster format. Actually, this is the poster presentation.
Actually, in my email, I got information about the oral presentation. That's why I prepared in this way, in not poster form.
Okay.
Okay.
One question is allowed. Thank you. Now moving to our next participant.
Thanks.
You please come forward. Sikhasri
Kunti.
Kunti.
Good evening to all. Myself Kunti Tandi from BRC. This project is related to radiosensitization. Compared to normal cell, in cancer cell line, RTK get dysregulated. In various studies, it was found that RTK help in radiation survival. So we hypothesize that targeted therapy using TKI inhibitor in combination with radiation has better control over cancer, and the efficacy of this treatment is depend on various intercellular signaling modulator. So for this project, I have 3 objectives. The first objective is to identification of CMET dependent human cancer using patient survival data, then study the effect of gamma radiation on CMET, then mechanism of CMET inhibitor induced anti-cancer efficacy. From the patient survival data, PDAC and glioma is found to be most MET dependent cancer. So for this study, we have taken 2 cell line for PDAC and 2 cell line for glioma. PDAC cell line is MIAPACA 1 and PENC 1, And for glioma, we have taken LN229 and U87mg. This Western blot data also showed that gamma radiation increase and activate CMAT. So then we studied anti-tumor efficacy using various TKI, and all studied TKI decreases cell proliferation, cell viability, and clonogenic survival of the cell. Among studied TKI, crizotinib is found to be most effective in PDSC compared to glioma. Crizotinib also causes cell cycle arrest and cell death. Crizotinib decreases phosphomat and its signaling effector molecule more significantly in PDSC compared to glioma. So we investigated the mechanistic insight for their differential sensitivity to crizotinib, and from the comparison study, we found that the intercellular signaling modulator are playing a very important role in their sensitivity. Use of then combination of crizotinib with ERK and beta-catenin inhibitor has significantly enhanced the crizotinib therapy. And crizotinib also increasing, it enhanced the radiation therapy. So in conclusion, PDSC and glioma is found to be most CMAT dependent cancer, and crizotinib is found to be most effective one, and the efficacy of treatment means beta-catenin and ERK is very important. They are playing very important role in the determining the efficacy of crizotinib therapy. Crizotinib enhance the radiation response. Thank you, everyone.
Question? Thank you, Dr. Tanti. Now,
Sikhasri.
Good evening, everyone. My name is Shikhashri. I am from BRC, and I'm here to represent my work. RECUL5, a DNA repair protein, modulates radio response through regulating pink Parkin-mediated mitochondrial dynamics in cancer cells. The major issue with radiotherapy is the development of radiation resistance in cancer cells. Many reasons have been postulated for this, but the role of mitochondrial dynamics in it has gained a recent attention. Talking about radio resistance, I would like to share a very novel and a very interesting results that we have found in our lab, that RECUL5, upon the knockout of RECUL5, which is a DNA repair protein, show radio resistance. To address this, the rationale designed is the role of RECUL5 in mitochondrial dynamics in response to radiation. The methods employed to achieve these rationales were clonogenic assay, immunofluorescence, live cell microscopy, seahorse experiment 10, Western blotting, plasmid reporter assay, among others. What we observed that in the clonogenic survivability assay, upon exposure to 4-gray radiation, that only 10% of the wild type means RECUL5 positive cells are surviving, whereas the survival rate increased up to 30% in the knockout cells, clearly showing the radio resistance of RECUL5 knockout cells. Since RECUL5 is a DNA repair protein, we wanted to check the DNA damage and repair kinetics. To do that, we did a gamma H2X foci assay, and to our surprise, we found that the DNA damage and repair kinetics was almost similar in wild type and knockout cells. This challenged us to look beyond the DNA damage, and therefore we moved towards the mitochondria. The first experiment we did was to check the mitochondrial membrane potential. For this, first we checked the mitochondrial membrane potential. We found that even after 48 hours of radiation exposure, the wild type cells are not able to regain its potential, whereas the knockout cells regain it significantly. To further dig into this, we did a seahorse experiment to know the basal mitochondrial respiration functional difference in wild type and knockout cells. we found that the basal mitochondrial functionality differs, giving wild type a positive, higher end in this data. So what could be the reason that even if the mitochondria is higher in wild type cells, it is not able to maintain its integrity, whereas the knockout cells can maintain its integrity. So to address this, we did a plasmid reporter assay where we used COX-8 as a marker for mitophagy, and we surprisingly found that the the events of mitophagy are occurring more in the knockout cells. In line to this, we did Western blotting to check the levels of the mitophagy proteins such as PINK1 and Parkin, and to our expectation, we found that the PINK1 and the Parkin localization was more on the mitochondrial fraction of knockout cells as compared to wild type cells. But to our surprise, the PINK1 protein in the whole cell extract was more in wild type cells. This explains the differential stabilization of PINK1 and Parkin in the mitochondrial fraction of knockout cells. We also did check the level of DRP1, which is a fission protein, and we found that the level of DRP1 was found to be more in knockout cells. From this, we can safely conclude that the radioresistance in Requel-5 knockout cells are not attributable to DNA damage. Rather, the efficient removal of damaged mitochondria through PINK1 Parkin-mediated mitophagy and the DRP1-mediated mitochondrial fission could be the reason behind. This is the concluding diagram that has been made, that upon radiation exposure, Requel-5 knockout and wild-type mitochondria behave differently. In knockout, the localization and stabilization of PINK1, Parkin, and DRP proteins are higher, leading to efficient mitophagy and helping the cells to survive. On the contrary, the wild-type cells, the stabilization is less mitophagy, and cells are not able to survive because of non-efficient mitophagy. Thank you everyone.
One question.
Very good, interesting work, and for practical application, how do we, clinicians, use this? Even if you have a side lab, or even if you have a lab in our hospital, how do we use it in clinic?
Sir, in patients, we have seen that the level of Requel-5 is, in some cancer patients, the level of Requel-5 is quite low. So we are more interested in digging into what could be the reason behind why these people not responding to radiation. This could be when we understand the mechanism, we could employ any drug or any treatment regime to treat such patients. First, we should know the mechanism why these Requel-5 knockout or knockdown cells are having an upper hand and are not responding to radiation.
Can we assess? We can assess in our lab.
Sorry, sir.
Can we assess in our lab, or only in the research center it can be done?
Sir, from abhi tak, I think it's in the research, but I'm very much interested in doing it I'm bench to bedside.
Thank you, Dr. Shikhashri. Our next is Dr. Ambika Sandalia.
Very good afternoon to one and all present here. I am Ambika from Naipur, Hajipur, and today I am here to present my research entitled as Mitochondrial Energetics and Altered Energy Metabolism Following Cranial Irradiation. This work was conducted in collaboration with Mahavir Cancer Sansthan, Patna. As we all know that radiotherapy is a primary strategy which is basically used for cancer treatment, and it leads to significant cognitive impairment in up to 75% of survivors. So we basically saw that radiation can lead to impaired cognition through the disruption of hippocampal cell proliferation, decreased neurogenesis, and increased neuroinflammation. However, the underlying molecular mechanisms are still unexplored. So to this, we postulated that how radiotherapy leads to cognitive impairment. We went on to find out the molecular mechanisms, that how it leads to that. So we went for acute head-only radiation of 9 gray, and radiation was given at the dose of 1 gray per minute to male mice. After 4 weeks, we checked out the level of various transcriptomes and the proteomes, like how the dysregulation of transcriptomes as well as the proteomes are responsible.
So we found out that we went for a transcriptomic analysis where we did a detailed mRNA sequencing analysis, and we found out that the genes encoding for various mitochondrial energetics related pathways as well as the neuroimmune modulation and mitochondrial energetics and glycolysis were disturbed. So we found out that basically the genes related to cognition like NSPSR1 and the mitochondrial energetics as well as the epigenetic genes were highly dysregulated. So after checking out these the gene level things or the mRNA level things, we went with the high resolution mass spectroscopic analysis to find out what all proteins gets dysregulated. So whether this thing is taking place at both the gene level as well as on the protein level. So basically the approach was the entire molecular level. So whether this thing, whether the entire molecular mechanism is involved. So we found that post the proteomic analysis, we found that the proteome analysis of the irradiated mice brain showed that various proteins related to mitochondrial energetics, energy metabolism, epigenetics, as well as the memory and cognition were highly dysregulated. So then we went on to collaborate both this data. So we correlated the entire data to validate the regulatory things, and we found out that we basically found out four proteins, the four genes that are basically responsible for that. So the four genes that we found out were the CRCTC1 as well as FAB7. And this study, after the correlation of the entire transcriptomics and the proteomics data, we further validated this using a Western blot analysis, and to our surprise, we found out that these proteins or these genes, these proteins were highly dysregulated, and so we checked on the impact of the glutamate-glutamine cycle, so whether radiation has any kind of impact on the glutamate-glutamine cycle and whether this is leading to persistent cognitive impairment. And we found that the level of certain neurotransmitters like glutamine, as well as the GABA, that is gamma aminobutyric acid, are highly dysregulated in the irradiated mice brain as compared to the non-radiated mice brain. Then we further went on with the immunohistochemical analysis, and we found out that the irradiation altered the level of various glucose transporters such as GLUT5 in the microglial cells, and this was confirmed by immunohistochemical analysis as well as the immunofluorescence analysis. So to conclude this, we can say that irradiation basically disrupts the tricarboxylic acid cycle or the Krebs cycle by upsetting the levels of various genes or the enzymes related to energy metabolism, and our integrated transcriptomic analysis, that is the mRNA analysis as well as the protein analysis, it together showed that various enzymes related to glycolysis, the Krebs cycle, the cognition related genes as well as the epigenetic markers were dysregulated, and finally we found out that cranial radiation decreases the glutamine level. Thank you, Radiation Research Society, for providing me the platform to present my research.
One question from floor.
Yeah, this is very interesting poster. So I wanted to know, since if you are doing cranial radiation only, so how much shielding we are getting to the other part of the body of animals?
So the entire part of the brain, we gave only head-only radiation. With the help of a collimator. There was a collimator, and radiation was given only to the part, to the head-only region, and the other parts were shielded.
Shielded, so that is how much shielding we are getting. Whether suppose you give 1 gray,
yes,
so how much other parts of the body they are receiving radiation? That is one thing. If you are not calculated,
the radiation was at 9 gray, and it was given at the rate of 1 gray per minute.
No, that is fine. What I'm telling that what shielding. So if you do shielding, how much like shielding we are getting other parts of the body? So that you can calculate. If you have not calculated, that is important. Second thing, it will be very interesting that if you have done cranial radiation, the another part actually which I'm going to also talk about are scopal effects. So if you do head radiation, what happened to other parts of the body? And we have published one paper some time back where we radiated only head of the rat.
Yes.
And then we looked the effect of the liver, and we found that liver metabolism gets changed. So that if also you can try to, since already we have got mice,
yes,
you are going to throw out other organs, so you can do some other organs. That will be very interesting.
Yes, sir. This could have been done, but we focused our research only to the cranial, like what
we can do now.
Yes, sir. Yes, sir. I understand. Sure. Thank you, sir.
Our next speaker is Dr. Shivani Behra.
Respected teachers and my dear friends, today standing before you is Shivani Behra, having a golden opportunity to give a presentation on the topic cordycepin, a derivative of nucleoside ameliorate, adenosine ameliorate neuroinflammation, neurodegeneration, and cognitive defect in irradiated mice brain. So, as we all know that due to radiation, there are cognitive injury and head damage. So, cordycepin is a drug. Cordycepin is a 3-deoxyadenosine derivative. The nucleoside that is a nucleoside adenosine we hypothesize that it can ameliorate neuroinflammation, neurodegeneration, and cognitive defects in irradiated mice. So this is my objective. So my first objective is to ascertain how cordycepin affect neuroinflammation and how cordycepin protect our brain from neurodegeneration and how it protect from cognitive defects. So this is coming to this is my experimental design. When we have 9-gray radiation to in cranial radiation, and we have treated the mice with cordycepin 50 mg per kg per day for 7 days long, and after that we have checked for different behavioral analysis, and after that we have sacrificed our mice for normal control and paraformaldehyde perfusion. And coming to my result part, we have not that is novel object recognition test to test hippocampal dependent memory, and we found that cordycepin treatment can increase our hippocampal dependent memory, as well as we did temporal order test to check recency memory and temporal order recency memory, and we found that cordycepin again can increase temporal recency memory in animal, where and for to check depressing like behavior in animal, we treat animal with cordycepin, and as compared to our disease control group, cordycepin treatment animal, so better result in force swim test, and for special learning memory, we checked by water maze, and we saw that cordycepin treatment mice has give significantly better result than disease control group, and we also check the level of we check the level of TNF alpha and beta galactosidase for neurodegeneration level, and we find that the 9-gray animal that are having more expression of neuroinflammation and cognitive defect, whereas cordycepin treatment can reduce neuroinflammation and increase cognition in animal. And we have also checked immunohistochemistry of GLUT3 and GLUT1 receptor, and we found that our cordycepin treatment increase GLUT3 and GLUT1 receptor. That is very important marker for blood-brain barrier, that cordycepin can protect our brain from BBB from damage. And so this is my conclusions are cordycepin protect our brain from cognitive defect, and it also and the expression of beta galactosidase and TNF alpha, it also protect our brain from neuroinflammation. Thanks to everyone.
One question. Thank you, Dr. Shivani. Our next presenter is Dr. Vishal Chabra.
So very good morning to all of you, very good evening to all of you. My name is Vishal Chabra. I am pursuing my PhD first year from Icrr, Hajipur, under the supervision of Dr. Vipin Parihar. I am here to present our work titled Biological Profiling of Astrocytoma using Liquid Biopsy and Exploring the Anti-Cancer Potential of Citrulus colocynthis extract in a glioma rat model. Gliomas, particularly astrocytomas, are highly aggressive brain tumors with limited therapeutic options due to their inherent resistance to conventional treatments like chemotherapy and radiation. To address the challenge, our study focused on two key areas. First is utilizing the liquid biopsy and proteomic analysis to identify various potential biomarkers for glioma detection and progressive monitoring. The other one is investigating the anti-cancer properties of Citrulus colocynthis extract in a preclinical glioma rat model. So coming to the proteomics part, here we have collected the liquid blood samples from the patients, and then we have done the proteomic depletion for the quantification of the depleted proteins, and then the content of the protein was estimated using the BCA assay, and further we analyzed this using the HRMS facility in Aiims Hajipur. From there we got results as a total of the 398 proteins were upregulated and downregulated which was shown by a Venn diagram, and in the volcano plot we have shown the upregulated proteins by the red color and green regulated proteins, green color with the downregulated proteins. So coming to the various significant proteins, we got only 39 proteins which are varying significantly in these results, and out of these total glioma specific proteins, or we can say tumor specific proteins, we got 5 to 6 specific proteins that are MMP2 protein and cadherin 13, spark-like protein 1, and protein S100B, and tubulin beta chain acids. So these are our findings in this, and further we'll go for the transcriptomic studies, and we'll collab these studies together, and will lead to the discovery of some important biomarker for the discovery for further analysis. And coming to the preclinical studies, here we have prepared the extract of Citrulus colocynthis drug. Here we took the drug concentrate and prepared its hydroalcoholic extract by using 50% methanol, and then we centrifuged it, and using the rotamapper evaporated it and converted to dry powdered form, and this dry powdered form then used to inject in the animal, used to given to the animals orally. Here we use the 4 groups. First one is a vehicle control, and then given to the then the disease group, in which we have performed the stereotactic surgery, and in the third group we gave our Citrulus colocynthis group after injecting the C6 cell line. Then the other one we have taken the standard group, in which we have given temozolomide drug as a standard. Then in the fifth group we given the combination therapy of CC plus temozolomide group. And coming to the in vivo imaging, here we performed the in vivo imaging using IVI fluorescence probe. Here we inserted the IVI fluorescence through tailwind, and we got various results in which the angiogenesis and vascularization was limited in the CCA group as compared to the disease group, and it was also limited in the CC plus temozolomide group as compared to the disease group. Then further we isolated their brains, and we kept it for further analysis like Western blotting and IHC. And then coming to one, we have performed one cell scratch assay, in which we have grown C6 cell line in our in vitro lab, where we have performed the experiment of cell scratch assay. Here we have used 250 microgram and 500 microgram of cells of CC, and coming to the conclusion, we got results in liquid biopsy and proteomic analysis offer valuable insights in the specific proteins. Then Citrulus colocynthis extract demonstrates strong anti-cancer activity, reducing tumor growth and cell migration in vivo. These findings support further exploration of CC as a complementary therapy for gliomas. Thanks.
Thank you, Shabra. No question, time is over. Now moving to our next Devidas Lemga.
Good morning everyone, myself Devidas Lamge. I am currently junior research in Aiims Hajipur. I present my work on dicycloserine partial NMDA receptor agonist. It's enhance fear extinction, learning and cognition in irradiated brain. Mainly here we are used as a dicycloserine, which is a NMDA receptor agonist, which used in the radiation induced radiation in radiation. We are given 9-grade radiation, and our main purpose to the explore the dicycloserine as a point of view of if effect on the mice behavior, mainly on the learning and cognition. So firstly, dicycloserine, it is a structural analog of D-alanine, and which is facilitated to adult neurogenesis and synaptic plasticities. It's appear and resolve the certain radiation complication in the CNS. And as a literature, dicycloserine is improved neurogenesis by acting on the spatial agonist in the NMDA receptor, which is the synaptic plasticity, and it's a critical process for learning and the memory, and thereby promoting the formation of new neuronal connection and potentially stimulate the neurogenesis in specific brain, which is associated with the learning and memory, particularly in the hippocampus. And hypothesis of study, it is a dicycloserine, it is a NMDA receptor and its agonist in a mood and memory, and it's trigger hippocampal neurogenesis and reduce the inflammation, like the marker of the CD11 and HMGB1. In the radiation induced cognition impaired memory. And studying the two objective at first objective is to evaluate the role of the dicycloserine in mood and memory, and second objective is to determine the effect of the dicycloserine against the radiation induced, mainly studying in all the study in all the behavior study and immunohistochemistry. So firstly, we have developed the experimental model using the 9-gray radiation, 1 gray per minute, an animal divided into 4 groups. First group is no radiation as a control. Second group, radiation with the treatment of dicycloserine. Third group is there are no treatment, only radiation group. And third one is fourth one is radiation with the treatment of dicycloserine, and cycloserine treatment given from the 7 day to 14 day. And here it is a fear condition test, and radiation group, third group is a 9-gray, showing the there are reduced time spent in the open arm, and similarly entries are reduced in the open arm. Similarly, there are showing the EPM and showing that decrease in anxiety, increase in anxiety in radiation group, and similarly we are also perform the FST. FST showing the time spent is immobile time increases in radiation group, and we also stain the immunostaining like HMGB1, and which is HMGB1 and NUN. This one is NUN, and expression of NUN is significantly reduced in radiation group, and similarly CD11 expression is increases in radiation group. And conclusion of dicycloserine mitigate structural damage while enhancing mood and memory impaired in mice in radiation.
Thank you. One more question. Now moving to our next presenter.
Thank you.
Dr. Himansi.
Good evening all. I'm Himansi. Basically I'm from Central SS College, and I have worked in my MSc dissertation at Bhabha Atomic Research Center research center, which I'm going to present here. So, other electron are used to treat cancer, but their biological consequences are unknown. So I worked on the project biological effect of other electron emitting radiopharmaceutical, a comparison of iodine 125 rituximab and iodine 125 deoxyuridine. For this, we first label the rituximab and 2'-deoxyuridine with iodine 125 through iodization method. In this process of iodization method radio labeling, we get that more than 90% labeling found of rituximab and only 4% labeling found for 2'-deoxyuridine. Then moving with the cell binding and inhibition studies, I found that the approx 10% binding on Raji cell expressed CD20 antigen, and more than 50% inhibition reached when inhibition test was run. Then we treated the Raji cell with the labeled rituximab and 2'-deoxyuridine for 48 hours and then performed various assays to detect the biological effect of other electron. So in the process of detecting biological effect, we first tested MTT assay, where we found that more viability found in labeled rituximab and less in more viability found in labeled rituximab and less in 2'-deoxyuridine. And for next with the LDH assay, we tested the membrane integrity of the cells. In membrane integrity test, we found that the 18% of LDH was released in medium in labeled rituximab test, but no LDH release found in 2'-deoxyuridine. Then we move with the MDA assay. In MDA assay, we detected the MDA is the end product of the lipid peroxidation. In this test, we tested for the oxidative stress and we found the oxidative stress in labeled rituximab. Then moving with the SOD1 assay. SOD1 assay, we tested for the detection of antioxidant defense mechanism, and we found the more antioxidant defense mechanism in labeled rituximab than as compared to 2'-deoxyuridine. Then with the 4-HNE test, 4-HNE is the byproduct of lipid peroxidation. It attacked on the carbon-carbon bond of the membrane, and we found that here it is the result of 50 microcurie treatment, and it is a dose dependent kind of, and in 100 microcurie test we found that the labeled rituximab is showing more for HNE ash. And ceramide test we detected for the labeled rituximab and 2'-deoxyuridine, and we found that the ceramide is more in 2'-deoxyuridine. And then we tested flow cytometry, performed flow cytometry analysis for the detection of apoptotic cells, and we found that in 2'-deoxyuridine there is more apoptotic cell than labeled rituximab. So at the end we concluded that comparing all the results, we concluded that the labeled rituximab is causing cell death through the lipid peroxidation, and 2'-deoxyuridine, which is a base analog, causing cell death through DNA damage. And at the last, I want to thankful for Radiopharmaceutical Division at Bhabha Atomic Research Center for providing necessary facility to carry out the project. Thank you.
Our time is over. No question is allowed. Thank you, Dr. Himansi. Our next presenter is Dr. Aliza Rehan.
my name is Aliza Rehan. I'm a junior research fellow at NMAS DRDO Delhi. So the title of my project is Unraveling the Role of Pheroptosis in Mediating Radiation-Induced Injury and its Crosstalk with NRF2 Signaling. So we all know that there are several types of cell death that take place post-radiation exposure, and pheroptosis is one of them. It is a form of iron-dependent regulated cell death that is characterized by the increased level of iron and lipid peroxidation. The role of pheroptosis in radiation-induced injury remains an area of active research with potential implication for radioprotective strategies. Our hypothesis is that pheroptosis, a major radiation-induced cell death pathway, is regulated by NRF2 signaling, which modulates the expression of key pheroptotic genes. So the material and method is as follows. We perform bioinformatics analysis, and after that we found out that pheroptosis was one of the key radio-responsive pathway that was activated. To validate it in an in vivo model, we perform survival studies with ferrostatin 1, which is an inhibitor of pheroptosis. We perform some biochemical assays for MDA, GSH, and 8-isoprostane. MDA and 8-isoprostane are basically the end products of lipid peroxidation, and GSH is glutathione, which is usually downregulated in case of pheroptosis. We then perform the qRT-PCR analysis and immunoblotting of the major 4 genes that we selected. So here are the results. These are the volcano plots depicting the upregulated and downregulated differentially expressed genes. The heat map was also constructed, and the Keck pathway enrichment analysis was constructed that denoted that pheroptosis was one of the key pathways that was being activated in both the data sets. So for the in vivo validation part, we perform survival studies for the mice exposed to 9-gray radiation, and we observed a survival rate of 16% after treatment with ferrostatin 1. We also checked the status of biochemical markers, and we found a significant downregulation of glutathione at 9-gray radiation dose, and significant upregulation of the end products of lipid peroxidation, that is MDA and 8-isoprostane. These levels were restored back to normal after the treatment with ferrostatin 1. After confirming that pheroptosis is indeed involved in the radiation-induced injury, we checked for the hub genes, and we identified top 15 hub genes, out of which we selected the 4 hub genes, out of which 2 were the promoters of pheroptosis and 2 were the suppressors of pheroptosis. The promoters ACSL4 and ALOX15 were selected, and the suppressors GPX4 and SLC7A11 were selected. We performed mRNA analysis and immunoblotting of these 4 genes, and we found that at 9-gray radiation dose, the suppressors were downregulated and the promoters of pheroptosis were upregulated. Whereas in case of ferrostatin 1 treated group, these were brought back to normal. Here is the RT-PCR analysis, and after that, to correlate this data with NRF2 signaling, we performed immunoblotting of NRF2 and NQO1, which is the downstream target of NRF2, and we found that at 9-gray, both of these targets were downregulated. And then in the ML385 treated group, the NRF2 levels were also downregulated, and it suppressed the functions of GPX4 as well as SLC7A11 and upregulated ACSL4 and ALOX15. So to conclude this, we conclude that pheroptosis is one of the key pathways that is involved in radiation exposure, and the key suppressors were downregulated and the promoters were upregulated, and NRF2 also plays a major role in mediating this pathway. Here are the references, and I would like to acknowledge Director Sir, HOD Sir, my supervisor, and my lab mates. Thank you so much.
Thank you, Dr. Eliza, for nice presentation, but time is over, no question is allowed. And now poster presentation for this session is completed, and it's a time for high tea, which is followed by the next session. Congratulations to all poster presentator.
Thank you, Chairperson. Thank you, participants, for the wonderful presentation. Now we'll break for tea. and we'll resume our second session after 15 minutes, to be chaired by Dr. Sudhit Chandana and Dr. H.N. Divakar. Thank you.
Presenters of the poster, please stay back. You are requested to come near the poster. Please kindly assemble. And one more announcement. At the end of the day, there will be a group photo session. I would like everyone to be there, please. Thank you.
Further delay, let us proceed with the next session. Session 2 on radiation protection and radiation reaction counter measure approaches. To chair the session, I would like to call upon the stage Dr. Sudhit Chandana, Head, Institute of Nuclear Medicine and Allied Sciences, Defense Research and Development Organization, Division of Natural Radiation Response Mechanisms, New Delhi. Together with Dr. H.N. Divakar, former Director-in-Chief and Consultant Orthopedic Surgeon, former President Orthopedic Society, Patna. Please join us in welcoming them. I would request the chairpersons to take over the session and invite the speakers as well.
Can you share a copy of the program?
And the session 2, which is on radiation protection and radiation countermeasure approaches. So we have chairpersons Dr. Chandana from Aiims Delhi and Dr. Nils Cordes from Germany. Just he has recently joined us. So we are going to begin this, and yeah, please.
Thank you very much, and very good evening to all of you. I think in this session we have 5 talks listed, but there are 2 speakers not present. And Dr. Amit, if we can just confirm, we have 3 talks in this.
There are Dr. Sunki Jo could not join us yet, so there are 4 talks scheduled.
The last one is by Professor Anjay Walchik.
This talk will be online.
Okay, his talk is scheduled online.
Yes.
Okay.
So only Dr. Sunki Jo is not
All right.
request for the schedule. Thank you.
Okay, thank you. So I have with me Professor Nils Cordes here, and both of us are going to jointly chair the session. And this first speaker is Professor V.K. Singh from AFRI, USA. The talk is titled Specific Biomarkers at Pre-Terminal Stage of Acute Radiation Syndrome in Non-Human Primate Model. So may I request Professor V.K. Singh to come and deliver his talk, and I think Professor V.K. Singh would like to deliver the talk while sitting because he has some health issue. The speaker may need a mic here and the control also, if you can help him.
Good evening. I'm sorry that I have to change this plan, though yesterday I was able to manage, but today I'm not able to manage, so please excuse me. First of all, I'm very much thankful to organizers for inviting us and providing wonderful hospitality as usual. So I'm really thankful. I'm going to talk about biomarkers. During yesterday talk, majority of you were there, and I little bit touched the topic that we develop radiation countermeasures and we develop biomarkers also, because development of biomarkers, identification, validation, integral part of radiation countermeasure development, because these radiation countermeasures get developed by US FDA rule, and for that we need biomarkers to convert the dose from animal model to human, because efficacy is not tested in human. We work with large number of countermeasures, and particularly we are focusing for radio protectors these days, because there are multiple radio mitigators approved for HARS. Yes, for GIRS there is no approval for any radiation countermeasures, so that also we focus. So this is just to show the importance that there are several agents which we work, and identification and validation is integral part, and we are working with biomarkers for combined injury also, in addition to HARS and GIARS. And radiation exposure dose assessment, for that also you need biomarkers. Although I don't work in that area, so my biomarker assessment identification is not for biodosimetry person. It's specifically for drug dose conversion under FDA animal rule. These are the biomarkers overall used. Whatever I have put in blue font, that I do, my lab does, specifically for radiation countermeasures. And here I will be presenting data for two, metabolomics and proteomics, not anything else. But all these we do actively. In metabolomics, whatever data I am going to present, that is only with blood. We have used serum as well as plasma samples of irradiated NHP, and we have done metabolomics and proteomics. You see, biomarkers in pre-terminal moribund samples of NHP. We are taking samples from animals which are going to die. We have written moribund. And we are going to take the sample very close to death, and that we call pre-terminal. So some of you may be wondering that if animals are going to die, they are very close to death, what is fun in taking their sample and analyzing the biomarkers, how it's going to help us in science. So that is for a specific purpose. Yes, it is very important, and particularly for us. We developed the drug for mass casualty scenario, and in any mass casualty scenario, it is not possible to treat everybody. So you have to triage that who needs treatment, who is going to get benefited by treatment, who is not going to need treatment because that person cannot survive. So you have to just take care those individuals for palliative care. So it is important to know that which exposed individual falls under which category. So this study is specifically for that we are interested to identify that these individuals are not going to survive and they don't need actual treatment, they just need palliative treatment. And by doing that, we can conserve our resources to provide to those people who really need that. So I'm going to present data of two studies. One, pilot study where we use 7.2 gray for identification of metabolomics and proteomics biomarker, and another study where 7.2 and 7.6 both dosage were used. Here for metabolomics data is published, I am going to present to you, and I am also going to present proteomics which is not published. We are writing the manuscript at that time. So in brief we will provide data for both with both doses. This 7.2 and this 7.2 dose is same, but it done few years apart, and it is just for the validation that it is not that whatever we are reporting it comes with one cohort of animal at one particular time and then later on we don't find it. And what we got the message out of that, we have identified several metabolites and proteins which can be used as biomarker for morbidity or mortality, and this is for triage purpose, and these findings will help to conserve medical resources. Before presenting the data, I will tell you that I was thrilled when kids were presenting their posters here for 4 minutes. Few of them presented that kind of data which I am going to present. Volcano plot, pathway analysis. So it was really fascinating for me that how young they are. They just join research into to do PhD, and they have knowledge of bioinformatics. They handle either independently or in group with lab members, everything which we do. So it was fascinating experience from that point of view. Experimental design for 7.2 gray metabolomics, and it is plasma. Another thing that we have done in plasma, we have also done in serum. So there is sample 7 days before radiation, 1 day after radiation, 13 days after radiation, 25 days and pre-terminal, where we declare that animal is not going to survive, we are ready to euthanize, and at that time just before euthanasia we collect the sample. So when we will make the comparison, we will make two different kind of comparison. One, this is pre-radiation exposure and these are post. So pre will be compared with post to show the effect of radiation exposure, and then post-exposure samples will be compared with pre-terminals to show that these are specific for pre-terminal state, and if those biomarkers are there, it means individual is not going to survive. Here we have given the number of samples, pre-terminal sample. It's difficult to get because what happens that animal gets sick and they die, and there is very complicated criteria when you declare those animals moribund and morbid. So here we had very limited sample, but in other study I will show you we have large number of samples, because either they are sick, they have not reached the stage of pre-terminal based on our criteria, or they die. So once they die, you cannot take sample from there. And that is the flow chart for metabolomics. This is, sorry. I did yesterday also same thing. Okay. On the top it is PCA plot, and this is volcano. some of them showed these volcano plots in their poster. So in volcano plot, what I am demonstrating, on left hand side it's negative, on right hand side positive, and there are 4 kinds of dot, and those are defined there. You pay attention to red, which is p value, it is significant, and log 2-fold chain also. So these are negatively regulated and these are positively. So it's PCA on volcano plot demonstrating metabolomic alterations in pre-terminal to post-radiation exposure. So you see here for all 3. This is day 1 versus pre-terminal, day 13 versus pre-terminal, day 25 versus pre-terminal. So you have lot of proteins here which you can work or we can validate with time. Here we have, those are metabolites, and those metabolites name we have given here. These are down regulated. And previous one. And these are up regulated. So we have selected few down regulated, few up regulated, and we will be working on those for further validation. Now, from same experiment here we have done proteomics and sample collection for 7.2 and 7.6. So it's similar a strategy, but this is experiment number 2 where 7.2 and 7.6 we have, and these 7.2 is not exactly the same. Means sample is not coming from the same anymore. Radiation dose is same, and there is at least few years gap between earlier 7.2 and this 7.2. And with this 7.2 there is another cohort of 7.6. And here you see sample collection 1 day before radiation exposure, on the day of radiation exposure, day 1, day 2, day 3, and pre-terminal. So we have selected samples which are little different in respect of time. And this is again flow chart of proteomics analysis. Just like last time, PCA plot and volcano plot. And here this is with 7.2 gray TBI. Day 1 versus pre-terminal, day 2 pre-terminal, day 3 pre-terminal, and this is volcano plot against negative and positive. This is for 7.6 gray. PCA plot, volcano plot, negative, positive, and again for same day, day 1 versus pre-terminal, day 2, day 3 pre-terminal. So there are large number of proteins which you can work with for validation. And these are down-regulated serum metabolites of 7.2 gray, and there will be down-regulation of 7.6 gray. Up and down-regulated serum metabolites for both. These are additional. So all these metabolites are there. So now these 2 studies with metabolite are over. Although sometimes I use protein, so that is not, that is just I misspoke. So these 2 studies were for metabolomics, and we used 2 different cohorts, and we showed that yes, there are a lot of metabolites which can be validated in pre-terminal sample, and that will be very helpful. So now plasma proteins profile in NHP exposed to 7.2 gray. And if you see, these are from same samples exactly where metabolites was done. So what we do, that we take archive samples and send it for mass photospectrometry, and wherever we send in the core facility, there same sample gets analyzed for proteomics and metabolomics. So that was for experiment 1, 7.2 gray. And this is again flow chart for that. It is just for comparison. That's what we are comparing here. PCA and volcano plots illustrating irradiation-induced proteomic dysregulation at day 1, 13, 25 compared to pre-terminal. Exactly same which we had done for metabolomics. And again here you see left hand negative, right hand side positive, and there are a lot of proteins which can be validated in pre-terminal compared to different day post-radiation, that is day 1, day 13 and day 25. These are box graphs to show certain proteins on different days. Day 7, day -7, day 1, 13, 25 and pre-terminal. This one integrin alpha 1, plasminogen activator inhibitor 1 and thrombospondin 4 and immunoglobulin lambda variable 11. So you see in -7 day post-irradiated and pre-terminal. So it is something really interesting. Something is getting up-regulated in that, something is getting down-regulated in pre-terminal samples. This is for proteomic study. Again, it is flow chart. It is coming from different studies, so we have given this again. PCA and volcano plot illustrating proteomic disruption 7.2 gray at these days. So it is from that cohort where we have 7.2 and 7.6 in parallel. And again, pre-irradiation versus day 1. So that's what initially I was mentioning, that we have done two different kind of analysis, pre versus post and post versus pre-terminal. So pre versus post day 2 and pre versus post day 3. And it is volcano plot again. And this is not published, this manuscript we are preparing. Now for 7.6 gray. PCA and volcano plot again you see, and with 7.6 also with day 1, day 2, day 3 and then pre-terminal. So this is for pre-irradiation day 1, pre-radiation day 2 and pre-radiation day 3, and then there will be pre-terminal. These are pre-terminal with different dose of post-irradiation. Day 1 versus pre-terminal, day 2 versus pre-terminal and day 3 versus pre-terminal. And you should see how many proteins you have which have been dysregulated at pre-terminal level. Same for 7.6, day 1 pre-terminal, day 2 pre-terminal and day 3 pre-terminal comparison, and this volcano plot. So take home message is there are large number of proteins to work with. This is my acknowledgement slide. These are my lab people currently. They have worked in recent past, and most of the studies are conducted in Georgetown in core facility. That is just that kind of facility where you can run mass spectrophotometry by paying the money. So we don't run in our lab. We don't have mass spectrophotometry. Although it is in our campus in university, we have BIC, that is core facility. But in my case, all my samples go to Georgetown University and mostly are analyzed by Dr. Amrita Chima, but I have few publications with Professor Al Fornes. That is academic collaboration, this is core facility analysis. And I get help from Dr. Rayan from PMB of our department for bioinformatics. And I have several grants with which these are supported because I do large number of proteomics, metabolomics, transcriptomic study. for biomarker for different purpose. Our Institute Director, Captain Burke, is here, and I'm also thankful for Radiation Source Department because that's something very cumbersome job for doing dosimetry for different cohorts of non-human primates. And Department of Laboratory Animals for vivarium access. And now I would like to have any question or query. I will be more than happy to clarify anything.
Any questions or discussion points?
Yes, Dr. Pandey.
Yeah, it is again.
No, we are not able to hear.
So very nice story again. New. I got two questions. One is that in one study you are using 7.2 gray and 7.6 gray,
Yeah.
two very close dosage.
Yes.
So what is the purpose of keeping these two dosages, and are you getting some difference? And the second question is that the pre-terminal, when you are saying, basically in that case the number of days may be very different from different animals, or it is going to be a certain, since pre-terminal is like just before death, or you did fix a date, number of date?
Okay. When we do non-human primate study, we don't do that study for one experiment for our analysis of something. No. Usually non-human primate study is done for survival, to test the drug. Now, from those animals we collect the sample and we archive it. So if you come to my lab, I will be having hundred of animal samples archived, because we don't know that what we will be needing tomorrow for what purpose. So all tissue, all blood samples, either serum or plasma, is stored archived in 4, 5, 6, -70 freezers. Now we decide. Question came that okay, we have to do pre-terminal, because it came from glucose. If you see blood biochemistry, we notice that when animal die, glucose level is very high. So that is separate story. From blood chemistry also we are identifying biomarkers. But we have omics platform, we decided that okay, let's one take dose 7.2 we took. And from that we analyzed and reported the result. Now we want to show that whatever we did, it is not cohort specific. And I will tell you that why we need to know that cohort specific. So we are going to show that it is not cohort specific. In that case, we cannot change the dose. We have to stick with that. So I had taken 7.2. Then in second experiment also I am pulling the same, because if I do multiple variation, it is not going to make sense. Now, next question is something only with 7.2 or it is applicable to other gray also? So due to that reason, we took another 7.6, which we are done together. We don't want to pull from different experiment. So due to these logistics, 7.2 and 7.6 we have taken. As far as days post-irradiation is concerned, we are interested to know that is it only works for day 1 versus pre-terminal, or even it works for 2 or 3 pre-terminal. So to show that in these two experiments we have selected blood samples of different days, not to same day. So means maximum variability we brought, whatever is possible, to validate our hypothesis that it is something really working, it is not a chance. Now I will tell you why it is this way. There is one German group, we collaborate with them extensively. It is German DoD Institution, Bandarware Institute of Radiobiology. Some of you attend Conrad meeting in their facility alternate year. They have large number of papers published with baboon, and they identified 4 genes that these can be used as radiation biomarkers. Yes, those studies were conducted in France. They got the sample because they can't do any animal study, and they identified 4 genes. Now when they took rhesus macaque, that is also non-human primate, they could not reproduce all 4 genes. And that research, they took it from one collaborator from California. After some time, they took it from me, my research, and now number of genes came further down. So it means whatever they got in one cohort of baboon that was not reproducible in research, and whatever they got in one cohort of research that was not reproducible in second cohort of research coming from different lab. So these are the issues we have to check to identify and validate biomarkers. So we keep everything in mind, and due to that reason we do. I hope I answered your both questions. And one more variable things we have taken serum as well as plasma, that it is not only with serum or not only with plasma, it works with both. Because in real scenario, you don't know that what is going to be available to you.
Yeah, thank you. Any other question? You can come forward first.
To know that whatever metabolites you have shortlisted as a radiation responsive metabolites, is there any possibility that these metabolites are also altered in any other conditions, any disease conditions or any other conditions?
Yes, some short-listed metabolites and proteins I had listed. It may be a small font, you may not have read, but yes, we have short-listed. At the same time, there are a lot which we are planning to shortlist. As far as those markers in another condition is concerned, yes, that is always a possibility. At the same time, we cannot do those conditions and we don't study those indications in our lab. So it is only going to be with literature. So yes, that will be also done in due course of time. Because whatever I am telling that these are pre-terminal biomarkers. So pre-terminal is one due to radiation, another may be due to malaria. With malaria also people die. Or autoimmune disease, that there is pre-terminal due to autoimmunity. So one has to check these that yes, it is radiation specific pre-terminal biomarker or it is across the board pre-terminal marker. But we are not very keen to get into that because that doesn't serve our purpose. Yes, that is academic research and it is always important. It may be important for some other group, but we are interested for radiation, and as long as we identify that person exposed to radiation having this biomarker is going to die, not going to survive, our purpose is fulfilled.
So thank you very much, Dr. Singh, for this great lecture and the questions.
Thank you for your attention.
So, first I'd like to say I'm very happy to be here. I'm very honored and also to immediately share a session. It's very spontaneously. So thank you very much for having me here. And so we come to the next speaker, which is Dr. Bhatt from Aiims in Delhi. And he will talk about interleukin-6, modifications of metabolic cell signaling to gain radio resistance and obviating radio protection. So we're looking forward to your talk.
thank you to the chair. I am grateful to the organizers for giving me this opportunity to present my work here on the platform of International Conference of Radiation Research, and especially thankful to Dr. B.N. Pandey because I came to Radiation Biology Conference after a very long time. So it is because he said that you must come, and then we should have discuss and then pursue that how we can take further the radiation biology work in future. So thanks to Dr. B.N. Pandey. And interestingly, when the chairperson has said interleukin 6, actually that was invited lecture 6, but my work also have interleukin 6. So that is a very nice coincidence that my work also has interleukin 6. So I'll be briefly discussing about it. So today I'm basically going to talk about modification of metabolic cell signaling to gain radio resistance and obviating radio protection. So basically what is our goal is mainly to protect being in defense. Like the major idea is like before in eventuality of any nuclear eventuality, when people are defense forces are entering into for the rescue operations, then we should have that kind of drug which they can take and they enter, so that they are sure that they are not going to die, the mortality is not there, whatever amount of radiation dose they are, up to the sublethal doses of radiation exposure they happens. So the major problem which happens is HARS and GIRS. Dr. V.K. Singh already talked about it. So the major problem which happens and why this HARS and GIRS is happening, I would like to briefly talk. I think majority of the audiences are already very well aware, so I'll be rushing through the introductory slides. That what basically happening here, that once radiation exposure happens, then it is not only the oxidative stress burst happens at that point of time. It is subsequently happen, and delayed ROS is also one of the things which is going to cause a major problem, and that is why probably a single dose of administration doesn't work, and we need to have multiple dose of administrations, and the strategy needs to change from there. So that is why, you can see here that again and again, and the basal level setting goes higher and higher, and that leads to more and more cell damage. That is leading to inflammation in the body, and that is what basically causing the major problem in HARS and GIRS. And people have tried lot of anti-inflammatory earlier. They thought that probably that can be a very good radio protector. But the problem what happened, like in inhibition of inflammation, because radiation induced inflammation which happens in the beginning is that itself is defensive mechanism of the body. So if we inhibit that defense mechanism of the body, probably we are going to sensitize it, and that is why probably it didn't work, the inhibition of inflammation before radiation exposure. And you can see here that when inflammation happens, proliferation is one important thing which happens, and proliferation is also driven by mild inflammation or pro-inflammation. Of course, not dysregulated inflammation, but mild inflammation is required to induce the proliferation, and that is what helps in recovery from the radiation-induced damage, and then the regeneration happens, and then proliferation goes down. So that is what the cycle, and there are many, it is very well established in the literature that inhibition of inflammation is basically, initially, inhibition of inflammation is not going to work as a radiation countermeasure strategy. So how the metabolism comes in picture? We all know that the inflammation is completely a metabolic driven process. Like what happens during the inflammation, NO production happens because of oxidative stress, and majority of the mitochondrial enzymes, they get nitrosylated, and in that nitrosylation what happens basically, that the mitochondrial enzymes stops working, and then glycolysis as a compensatory mechanism goes high. And you can see in the same thing happens during bacterial infection also. Glucose level in the blood goes high, and that is what basically providing glucose to more and more cells, leukocytes and several cells which are basically involved in protecting the body. And glycolysis is not only a bioenergetic pathway. People have already demonstrated that it is a survival pathway, and the cells which cannot induce glycolysis after radiation exposure or any kind of stress, they are very susceptible to stress, and those cells which can induce glycolysis, especially by involving this enzyme PFKFB3, they are able to rescue themselves from the stress, and that is why probably it works. And after radiation exposure, you can see here that on the left side of the cells, basically the cells, those which cannot rescue themselves, and on the right side of the cell, where after radiation exposure, the remaining cells which induces their mitochondrial biogenesis, they survive much better. And that is what you can see in the end on the bottom side, that huge amount of pharmazone conversion. A lot of students were presenting the MTT assay in case of radiation, and we published this data some 4 or 5 years back, that actually MTT assay doesn't work in case of radiation. Your results will be very erroneous. So when you are doing MTT assay, be very careful from that perspective. So basically this glycolytic upregulation, people had already demonstrated in case of cancer that it is mainly Warburg effect, that even if you take those hypoxic cells in the environment of oxygen, they will not be able to, they don't go back. They actually, use more and more glucose for their, for majority of the stress for their proliferation. And that is what an important process. As I discussed, that radiation-induced inflammation is a defense mechanism of the body, and induction of glycolysis is required for the inflammation, and inhibition of glycolysis, we already know that in cancer patients, we are already doing that for radiosensitizing the tumors. So we hypothesized that can induction of glycolysis results in radioresistance, and can it be radioprotective? So that approach we have taken, and we have shown that transient elevation of glycolysis confers radioresistance by facilitating DNA repair in cells. And we chose 3 molecules, DNP, PTEN inhibitor, and interleukin-6 for protection of the cells from radiation-induced death. So DNP is the first molecule. We already know that mild mitochondrial uncoupling is shown to be a very good protective agent, cytoprotective agent, in case of ischemic reperfusion injury, and there are a lot of literature evidence for that. And ischemic reperfusion injury, if you see the sequential events of ischemic reperfusion injury and IR-induced injury, they both are very common, and that is why we thought that probably mitochondrial uncoupling can protect. And you can see here that what we have done after we chose different mitochondrial uncouplers, and with that we did mild mitochondrial uncoupling, you can see here, and higher mitochondrial uncoupling both, and we saw that in mild mitochondrial uncoupling the oxygen consumption rate goes high. That means the mitochondria are not becoming non-functional. It is just that they mild mitochondrial uncoupling, it is regulating the oxidative stress and several other pathway more in more coordinated and regulated way. And once we did that, we found that the glycolysis in the cells has also gone up, and we found that through glucose consumption and lactate production. And you can see here that the total oxidative stress and mitochondrial oxidative stress, both in case of uncouplers treated cells, they both are very nicely, it has come down as compared to radiation alone. And you can see the cycle what I have shown in introduction in the beginning. We have followed that time kinetics of reactive oxygen species. Red line is the delayed ROS which is being produced, and once the cells were treated with DNP, that the green line, you can see that is very nicely coordinated, and probably that is why it is not leading to more and more cell death. So this is after DNP treatment. We checked in majority of the the cells which were available with us, hematopoietic cell and gastrointestinal cells, and they showed that good amount of radioprotection and very minimal cell death. And we checked whether calcium signaling is playing role here or oxidative stress. We used BAPTA-AM and as well as mitotempo, and we showed that it is not only BAPTA-AM or not only mitotempo are able to regulate. DNP, which is doing both, is providing much better radio protection as compared to BAPTA-AM or mitotempo alone. And the same thing was demonstrated with the help of DNA damage also. This is transgenic cell lines which forms 53 BP1 foci, and you can see here that it is very nicely bringing down the foci per cell. So overall summary of this DNP molecule is that it basically, DNP, because it brings down the mitochondrial membrane potential, what it does, like, it doesn't allow calcium to go and accumulate in the mitochondria, so it is not damaging the mitochondria. That is one thing. Then second thing, it is upregulating glycolysis as well as oxygen consumption rate, because of which the ATP production is much better, and that is what probably facilitating the DNA repair in much faster way, and it is able to rescue the cells. So then we tried this in animals, DNP in C57BL6 mice, and you can see here that we got around 40% change in the survival in mice. And very nicely you can see that on the left side, the hairs, I think probably because of the light, it is not very nicely visible, but the gray hair you can see in the radiation alone. But those animals which are treated with dinitrophenol, you can see that they are not, the gray hair is there, but it is not that much. And there is already in literature, it is demonstrated that keratinocyte stem cells, if they are protected nicely, then the hair graying will be much less. And probably then we thought that probably DNP is protecting the keratinocyte stem cells also. The second approach is the inhibition of P10, and you know that P10 is continuously dephosphorylating AKT and AKT's upstream kinases, and because of that it is not able to, AKT is not able to activate it. But if you use P10 inhibitor, then AKT signaling can be induced, and that leads to the increase in glycolysis. And we demonstrated here that increase in glycolysis using P10 inhibitor, it is again in those 3 cell lines, it is giving very good protection. And it is by inducing the AKT signaling. You can see here on the top left side, the western blots, the AKT phosphorylation is happening very nicely. And once and if you inhibit the AKT signaling, then whatever radio resistance we are getting, that gets abrogated. And if you silence the AKT also, then also P10 inhibitor mediated radio protection, it gets reverted. So that itself shows that P10 inhibitor is basically giving protection through AKT mediated signaling. And then P10, it facilitates DNA repair, and in the end you can see that the micronuclei has also gone significantly down. And then we again checked this. We injected P10 inhibitor intraperitoneally, and you can see that AKT phosphorylation is coming very nicely in PBMCs, gastrointestinal tissues, bone marrow and spleen. In all the cells, the AKT phosphorylation we are able to achieve, and it is leading to a good 20-30% protection, this molecule also. So these 2 molecules we have tested that by inducing glycolysis we are able to get protection. Then the third molecule is interleukin 6, and it is also very well known that interleukin 6 is known to proliferate the leukocytes, and it is also cytoprotective in many cases. It has mainly 2 different mechanisms of classical and trans signaling, and like when we are running also, it is known that the interleukin 6 level goes very high. It is known as a myokine, and it alters the metabolism of the muscle cells, and it gives protection there. So it helps the cardiomyocytes during that excess burden of the running or any muscle activity for the cardiac protection. The IL-6 level, increased IL-6 level is very much required there. So we have seen that IL-6 is able to increase the glucose metabolism. We have checked with different enzymes also, and then transporters also, and it shows that it is able to increase the metabolism. Because of the paucity of time, I could not bring all the data, but you can see here that this is our in-house developed assay where we can assess that the mitochondrial damage, and all the bodies you are seeing this, sparkling body in IR, you can see that is basically damaged mitochondria which accumulates a lot of A23187, and when the cells were treated with IL-6, those bodies are hardly you can see, but when you inhibit the glycolysis using 2DG, again you can see that the body formation starts coming up. So it shows that glycolysis is not only by facilitating DNA repair, it is probably able to protect the mitochondria also, and increased glycolysis is already known in the literature that the ATP which is coming from increased glycolysis is basically protects mitochondria as well. So here also we have seen that the cell death is significantly down, and that is by PARP cleavage and caspase 3 cleave. You can see that in the IL-6 treated cells, it has gone significantly down. And then it is mainly through STAT3 phosphorylation, and IL-6 basically induces STAT3 phosphorylation, and by that it is basically, facilitating the pro-survival signaling. And MCL1 and survivin also, we have seen the level of MCL1 and survivin has gone up. And you can see that the STAT3 phosphorylation in the top one in all IL-6 treated, it is gone, it is high, but not in other groups. And P53 also has gone significantly down, suggesting that this is helping the cells to recover from radiation-induced cell death. But when we used JSI, which is inhibitor of STAT3 phosphorylation, you can see that the whatever protection we were getting, that got reverted, and the cell death pathways also is again comes back in radiation-induced. And we have seen here in case of gamma S2X as well as P53 BP1 foci, that also has gone significantly down in IL-6 treated cells. So the summary is, by IL-6 basically by activating the antioxidant defense system, by facilitating DNA repair, and by facilitating pro-survival signaling, it protects the cells from radiation-induced damage. Then in vivo studies, we again injected IL-6, and we found that in PBMC's bone marrow and spleen, you can see that STAT3 phosphorylation is very nicely coming after 2 hours of injection, and then AKT phosphorylation is also coming very nicely, and this is giving much higher protection than the previous 2 molecules which we have seen, that you can see. And when we use JSI on the right side survival curve, you can see that JSI is able to abrogate all the protection which we have got in that. And IL-6 treatment, it protects the transverse section of the bone marrow, the bone, and the vertical section of the bone also. Both suggest that there are good amount of stem cells are protected. And the GI also, we have, these are some preliminary data. We found that the GI is also very nicely protected. And the message is, so this is the last slide. So the message is that metabolic modification, it reduces oxidative stress, reduces macromolecular damage, and facilitates DNA repair, and it protects the cells from IR-induced cell death, and it leads to radioresistance, which is ultimately giving us the radio protection. So this is the acknowledgement, Dr. Dwarkanath, who introduced me to this field of metabolic modification and radio protection. Dr. Chandna, who facilitated this work currently in the institute. He is currently director, and these are the students who did all this work. Yogesh and Ankit, they did P10 and DNP work, and Neeraj and Dhananjay, they supported this work. Thank you very much. If there is any question, then if chair permits, then probably.
Thank you, Dr. Bart, for this nice presentation. So we have time for one quick question. Quick question, please.
Yeah, great, very comprehensive presentation. I really enjoyed it. The question I have is, you talked a lot about glycolysis and how it promotes ATP, and then all the mitochondrial health and all that. Another way to look at the whole scenario is to consider non-glucose molecules. In fact, anti-glycolysis, if you will. Ketosis generates as much ATP. The advantage of ketosis, or ketone bodies, is that during ischemia, body really struggles to metabolize glucose. That's well documented. And we have done a lot of work. Much of it is patented, both in animals and in humans. Ketosis does several different things, actually. It promotes ATP, relieves ischemic issues. Yeah. And most importantly, for the defense people, it improves cognitive power, both in animals and in humans. But I haven't seen any publications, maybe defense people might know about it, that during radiation damage, giving humans ketone bodies. I haven't seen that. So can you comment in the light of, and again, ketosis will not induce IL-6. ketosis induces IL-12 spectacularly. We have seen that in small animals, medium-sized animals, and in humans. And IL-12 is great actually. That's really going to lead to, if there is a mitochondrial dysfunction, it's going to set the mitochondria right. So lots of advantages. Any comments on that?
Thank you very much. This is very nice suggestion what you have given. Actually, if you see the IL-6, I mean ketosis may not be inducing IL-6, but IL-6 induces ketosis. So in my summary slide, introductory slide in the beginning, you would have seen that that's how it modifies the metabolism. As I said, when IL-6 comes in serum, then the similar kind of scenario is there. I mean, basically, the insulin resistance which generates, which Dr. V.K. Singh was also telling, when the subjects are going to die, at that point of time, probably in this kind of scenario, sepsis is very high, and in that kind of scenario, insulin resistance will be there, and of course, glucose level will be very high. So the similar, situation happens in case of this also. Bacterial infection in the initially when happens, or even if you're giving interleukin-6. So what is happening, that the glucose level in the blood becomes very high. Because majority of our leukocytes or the cells which are regulating inflammation, they need either glucose or free fatty acids, right? So because majority of the macrophages through CD36, they basically take lot of free fatty acids. So that is why all this kind of adipose tissues, IL-6 works in adipose tissues also, and it makes lot of free fatty acid available. So that is what becomes the, raw material for some of these cells. So definitely what you are suggesting, I think that is in the line with whatever metabolic changes happens in the body. So definitely, if somebody is not trying IL-6, probably directly free fatty acid can be tried.
Dr. Bhatt, thank you very much.
Yeah.
So we need to move on. Thank you.
Thank you.
We have now the next talk is by Dr. Amit Kumar from BARC on nuclear radiobiology research, mechanism, guided decorporation, and mitigation approaches. Dr. Amit, please.
Yeah. Thank you, chairpersons, for your kind introductions. Let me thank local organizing committee to provide me the opportunity to speak our research in this field, nuclear radiobiology, and let me first of all little bit talk about this field. So this field deals with understanding the radiobiological effects of the radioactive material which have internalized inside the human system. And the second part of my talk is about how you can develop approaches for the removal and also the mitigation of their radiological and chemical toxicities. So let me begin with the next, yes. Hey, why didn't work? Yes. So let me start here with the, sorry. Yes, so let me start here with the climate change, which is a buzzword in general public. We all have started facing the worst effect, the effect of climate change in our life in terms of severe extreme weather. Recently, the COP29 meeting has officially concluded that if we have to prevent the worst impact of climate change, you need to achieve zero carbon emission. And that is possible by adopting the nuclear energy. That's what the IAEA has made the statement in the reports. Now, what I have, I'm sorry, yes. So, as far as concerning the nuclear solutions, we have the three nuclear fuels. Uranium we have been utilizing. Other options are thorium and plutonium, and these two are having much more quantum of energy and also offers sustainable means. Now I brought a table here And this table shows the release of nuclear materials from Chernobyl and Fukushima event. If you combine both, that means the risk of release of radioactive materials cannot be avoided if you pursue occupational, whether you pursue occupational practices from nuclear fuel cycle or it is the risk of nuclear accidents from your facilities. Now I'm here to share some of the milestone reviews and research articles from the field of nuclear radiobiology, which appeared in Nature, in Cell, in Science, and these just acknowledges the significance and relevance and importance of this field. Now, interestingly, interestingly, we have been organizing, the IARR have been organizing very dedicated conference to talk about the recent developments in this field, its health effects of internalized radionuclides. Yes. So globally, if you see, there are some reputed research centers and institutes which are pursuing nuclear toxicology research program. Just a minute. Yes. Sorry. Yes. So if you look at here, the institutes are pursuing nuclear toxicology research, having very dedicated facilities to handle these radioactive materials for cell and animal experiments. Also notice the significant contribution of AFRI for depleted uranium and cesium-137s. So, when we started our program, India also started contributing to the research of nuclear radiobiology with these countries. Yes, as I mentioned that we have been utilizing the uranium. This is the abundance of uranium in various countries. Now, but if you compare the abundance of thorium, it is 4 to 5 times much more abundant than the uranium. And that, if you look at its distribution, the distribution is also quite good among various nations. That suggests that the thorium has the potential to be a fuel of the future for sustainable nuclear energy generation. Now, to demonstrate that, to be future ready, India has started pursuing adapting thorium-based nuclear energy, and our center BARC successfully developed thorium-based fuel, and this is the 3-stage power program of India, in which thorium will be utilized in the 2nd and 3rd stage. And I must share a news that India has advanced the development of fast breeder reactor to the fuel loading stage. That means the commissioning of this reactor will open the gateway for large scale utilization of thorium. And in addition to these reactors, thorium-based fuels are now available to be utilized in PHWR and molten salt reactors. So these are in India, these are the thorium plants where we are handling this much 1,000 tons of thorium, which Saurabh also talked about in his presentation. And if you, in addition to India, other countries like Australia, Canada, Russia, they are also separating thorium from the mineral, and this is what the concentration of thorium they have reported in the blood of their workers. Also, likewise, India also reported the thorium concentration. So if you look at so you get a sense that the exposure of thorium cannot be avoided if you are going to pursue the nuclear fuel cycle thorium based. But now if you look at some of the documents from regulatory agencies like ASTDR from United States, this data it clearly suggests that not sufficient data is available to determine the permissible level or other regulatory limits for the exposure of thorium. Now, if I look at here, the ICRP or it is the UNSCR, they all talked about on the similar conclusion that there is insufficient data and nothing is not much known about the toxicity of this thorium actinide. In fact, they are also emphasizing that we need future research is needed. This is what the document we cropped out from the CEA France document. And so this suggests that we need to generate data and do the research in this field. So if I summarize the sequence from the exposure to the health effects, thorium gets absorbed in the blood, finally accumulates in these target organs like liver, bone, and also notice lungs is one of the important target organ in case of inhalation exposure during occupational scenarios. So when we were conceptualizing this project in 2004 at BARC under the guidance of Dr. K.P. Mishra, these two objectives were quite clear in our mind, that we need to understand the mechanism of radiological and chemical toxicity of this important thorium element. If we understand well, we should be able to develop the rational approaches, the biologically rational approaches for decorporation and mitigation. So what we did, we started our program by looking at the effects of thorium on human RBCs, human blood proteins. We extended our studies in the liver, bone, and also when Saurabh joined, then we looked at in case of the lung cells. So in next few slides, I am going to highlight the key mechanism about the effects of thorium in some of these biological systems. So if I in the next 1, 2, or 3, 4 slides, which I will be sharing with you. So in this work, what we did here, this is the work about the effects of thorium on human red blood cells, and so you can notice this is the whole blood experiment. Notice the thorium induces the leakage of hemoglobin in the blood plasma. However, uranium has not such effect, clearly suggesting chemical toxicity of both the important elements are completely distinct. Now we also took the other rare earth materials which are present in the thorium sand. They also do not have the effect except cerium-4. We also identified, this is the isolated blood experiment, and thorium was found to cause aggregation effect and also the lysis of the effects. And thorium does so because it is of highly positively charged metal, it's +4 metal charged, so it just binds the surface, the negative charge which is present on the surface of the RBCs, and this is how thorium targeting for the aggregation effect, that is the surface charge neutralization. And osmotic imbalance was found to be responsible for the lytic effect of thorium. So you might have noticed that in lysis tube, the color was quite unusual. It was little brown color, so we hypothesized thorium, I think, is affecting the hemoglobin. So in this paper we studied the effect of thorium on human hemoglobin, and what we found, thorium is affecting the heme component of the hemoglobin. However, other metal ions like uranium or lanthanum or other rare earth materials, they were found to interact only with the protein part of the hemoglobin. And our data suggests that thorium does so because its charge is to ionic radii ratio becomes very close to the iron. That's why it has this kind of effect. So this is our data about the effect of thorium on albumin, and here this data further suggests thorium binds much more high stoichiometry as compared to the uranium. And this is the work done by the Saurabh and in lung cells, which he has already presented in the Young Scientist session. So I would not talk in detail, but notice that thorium dioxide goes more as compared to the nitrate, and this is what he was talking about, thorium-induced DNA damage response. This is what it encapsulates most of his findings. So this is also, I think, Saurabh has talked about, and we are delighted to share some of these findings. It has the importance that this observation has the relevance to utilize the expression of SPD as a biomarker of thorium exposure under inhalation scenario. So this is what, and when Rocky was with us in our laboratory in 2018 and 20 during those, she identified that when thorium goes inside the liver cells, it is targeting the cytoskeleton as compared to the other subcellular sites. And this thorium does so because it binds to the calcium binding sites. So Suraki has also looked at this. She also validated some of these findings under in vivo conditions, thorium injected, and she found disturbances in the cytoskeleton and liver tissues. And this is the long-term effect of thorium. That means after 6 months and 1 year, and omics data concluded that thorium upregulates the carcinogenesis mechanism by engaging beta-catenin and MYC oncogene. This is the validation at RT-PCR and protein level, which both Saurav and Raki has nicely elucidated the concept, the possible mechanism of the carcinogenesis. And here, this is my first part. I will very quickly, I have hardly 3 minutes. So the effects we have used as an assay, and the mechanism we have used as a rationale. So what we did in this part, we know that we need to remove thorium from liver and bone, and this agent is available, but it is not effective from liver and bone. It just works only with the blood. So, and these limitations are very well reported. So in our laboratory, we have adopted 3 approaches. One is that we delivered this clinical antidote by liposomes to the liver and bone, and second approach, we rationally select the compound on the basis of understanding of the mechanism, and third approach is we also entirely synthesize entirely new agent on the basis of understanding of our mechanism. So I'm going to share the results on our second approach. So here in this paper, what we did, we have selected about 25 molecules on the basis of understanding of the mechanism. For example, iron binding sites or calcium binding sites or hepatoprotective agent from all the work that we have done in last about 10 to 15 years. So what we did here, we have screened about 25 compounds, and we compared the efficacy with the known DTPA. In all these assays, we compare the efficacy of these compounds with DTPA, and then we also characterize our compounds by complexation studies like molecular simulation or ECJAPS, experimental data, and finally we t-test in animal models. So what we did here, I'm going to see all this data we have generated, we have published. I'm going to share the results of hemolysis. It is highly observational, so you can clearly see that if your compound has higher affinity to bind thorium, it will cause less amount of leakage of hemoglobin. And that's what we generated the data. Here green number indicates these compounds are more effective than the DTPA, and this is I would like to have your attention that this is thorium-induced damage, and these 4 compounds have much better effect to prevent the thorium damage, and also found to be more effective than the DTPA, which is, notice here that DTPA could only prevent this much of damage. However, our 4 test compounds, rationally selected compounds, found to be more effective than the clinical DTPA at equimolar concentrations. We tested in animal models, so in metabolic cases, and here we tested this L52, which was the most effective in the screening. So we made 3, this is the metabolic cage experiment, having provisions to collect the urine to see the impact of our test and comparative therapies on the removal of thorium in the urine. And first we performed 3 group experiments. Later on we also repeated the experiments by adding combination experiments. I will tell you the reason. And if you look at here, this compound L52S, which worked so good in all our in vitro experiment, in vivo condition it did not work. If you look at the DTPA, which is like a test molecule, it works so good up to day 10, but beyond this it didn't work. We were just puzzled about it, but now we were knowing that the DTPA is highly hydrophilic. So we just combine, and what we noticed that interestingly L52 is able to mobilize thorium from some of these organs, liver, spleen, bone, but was getting accumulated in kidneys. So what we did, we combined, and what we found that this combination resulted more excretion of thorium through the urine. So this is the long-term experiment data. This is the mitigation of the DNA of the inflammatory markers.
Dr. Amit, you can
yes.
wind up soon.
In 30 seconds.
Quickly.
In 30 seconds. Yes. So this is the characterization of the complexation, and this is the last slide summary. And so green one indicates the target site of the effect of thorium or the processes by which it causes the toxicity in some of these systems. And this is quantitatively it is about 2.4-fold higher decorporation of thorium. This is I'm just humbled to share that our results are being accepted by some of these journals. And yes, there are some regulatory agencies like CA France have also started reporting some of our findings involving in formulation of policies by UNSCR, by IAEA and all that. And with this, I finish. Let me complete my duty of acknowledging the contributions. Let me express my sincere gratitude to Dr. Mishra, actually, who has conceptualized this program in 2003. And I also sincerely acknowledge the contribution of my team here, with Dr. Pandey is leading here, and Dr. Nina, and I've shown you the contributions of all the colleagues, but Dr. Nina and Dr. Basumati is also joining this project by studying the effect of thorium in combination with alpha radiations or delivering the nano formulations of DTPA. And I must address here the contributions of various divisions of VARCs to purify the thorium, to characterize and detect it, and also the other units of DAE. And let me finish my presentation here by dedicating this talk to Dr. Bam Bahadur Singh, who was the student of Dr. A.R. Gopal Iyengar. And Dr. K.P. Mishra was the student of Dr. Bam Bahadur Singh. I am just inheriting all the learnings from some of these generations. And with this, I thank you very much. Thank you very much for your kind attention.
Thank you, and thanks for reminding Dr. B.B. Singh. Thank you so much, and we all remember him. And any question? We can have a quick one question, very short one, please.
Yeah, great talk. I really enjoyed. As you nicely pointed out, the similarity of ionic radii of thorium with iron is a problem. In animal models, either your group or somebody else, have they shown that excessive thorium in the animal body would lead to anemic situations?
They make you?
Anemic situations.
Oh, yes. Very important observations. And it is our, let me first cite you that there is one reference that the erythrocytopenia, the decrease in the RBC count, was very well reported in case of Russian thorium workers. So that is the epidemiological data on thorium workers. But now let me cite here our own experimental observations that when we inject thorium at let's say the more than 1 mg per kg, because since thorium induces the aggregation of RBCs, if you inject thorium in the blood by intravenously at high dose, and this aggregation is causing the sudden death of the animals. So aggregations is also we have observed in in vivo conditions, and chronic exposure conditions, this anemic like or erythrocytopenia like conditions. Yes.
Is it time for another question, or we are done?
Well, we are only running short by 2 hours, but you can quickly.
Okay.
Please.
The observation you pointed out, DTPA is too hydrophilic.
Yes.
It cannot remove anything from the liver.
Right.
So for something to go to the liver, pick up whatever you need to remove from the liver,
yes,
you need an amphiphile,
yes,
molecules with the dual hydrophilic and hydrophobic characteristics.
Right.
Your observation is in line with what we have observed, and we can talk about. We have come up with some very interesting amphiphiles. They go to the liver, pick up what you're supposed to get out of the liver, and
yes,
clear through the renal system. We can talk about
yes,
that.
Yes, that's important observations. Actually here, when it comes to the concept of decorporation agent, these two properties are mutually exclusive. Means when you are making the molecule to make it more effective to decorporate actinides from blood system, that means it cannot access your organ systems. So somewhere you have to have the very right balance. And when we actually we tried delivering DTPA by liposomal systems to overcome these limitations of DTPA which you have talked about. But now our experience, when we compare the efficacy in all our studies, what we found that still combination of L52 with DTP plus DTPA, I mean oral L52 and intravenous DTPA, is still much more effective than the liposomal DTPA. And also when we compared our efficacy data with the hydroxypyridinate synthetic chelating agent, which is designed by LBNL by Professor Dr. Rebecca Groves, and when we compare the efficacy, in fact it is the statement from their publications that HOPO is not that much effective to remove actinides from the bones, but here this combination is still very effective. And also here deliberately we have delayed the administration of L52 after thorium contamination. It was after 24 hours of thorium contamination. That means you have allowed the absorption of thorium into the organs, and then you are testing your therapies. So okay. Yes, we can talk about that.
Yes. Thank you, sir. Thank you very much, Dr. Kumar, for this great talk.
So
we move on, come to the last talk of our session, and I'm happy to introduce to you Dr. Andrei Wojcik. He's talking to us online, and hello, Andrei. Good to have you here, and if you are ready, please start. We're looking forward to your lecture.
Thanks, but the lecture is pre-recorded, so you will hear it from the PC.
You're not speaking through AI, no?
No, I am not, but I sent a pre-recorded lecture, so it should be
great.
Presented to you in a minute. And I would like to thank the organizers for giving me the opportunity to speak to you today about the question of whether there is a genetic component of individual response to radiation. Now, if you read textbooks on radiation biology, like this one here, you will find the information that the quest for biomarkers of individual sensitivity can be described as the holy grail of radiation biology. So the question is, can we predict the reaction of an individual to radiation with respect to cancer effects or deterministic effects? Yes, we could in principle be able to do this if the reaction is driven by the genetic background of the individual. So let's have a look if there is any proof that the individual response to radiation is genetically determined. And the first thing that we can do is we can compare the sensitivity of men and women. And if we do this, we look at the risk of radiation-induced cancers among the survivors of Hiroshima and Nagasaki atomic bomb droppings, then we will see that indeed females are more sensitive than males. Here is the excess relative risk for all cancers as a function of attained age for different ages of exposure. And again, females are more sensitive than males, about twice as much. So that is a proof that there is obviously a genetic component in individual radiosensitivity. But what about if we look at people of same sex? Is there something that we could call the intrinsic individual radiosensitivity would be genetically determined? And again, the answer is yes. We have individuals with very rare monogenic diseases like ataxia telangiectasia, like Neymer-Head breakage syndrome, Fanconi anemia, or ligase 4 deficiency. And these syndromes are associated with severe lack of DNA repair enzymes and make the cells of these individuals very sensitive to radiation. You see, this is the surviving fraction following a certain dose of radiation, and this is the level of survival for normal individuals or for heterozygotes of ataxia-telangiectasia, ectasia, but homozygotes AT, NBS, FA, ligase 4 show a dramatic sensitivity, suggesting that yes, there is a genetic background to individual radiosensitivity. But what about people who have a normal phenotype, who do not show a monogenic disease? Can we identify them as, or can we identify people who are sensitive, more sensitive than others? And here we focus on this question, and we look at the reaction of cells and cells from individuals and cells in culture to mixed beams of low and high LT radiation. So the question for us, the interesting question for us is, how do cells react to a scenario like this, where they receive both scattered DNA lesions and DNA lesions that are densely packed along a track of an, for example, alpha particle? And we have two possible outcomes of combined exposure: either additivity, where the outcome is the same as predicted from adding the effects of these individual radiations, or synergism, where the outcome is stronger than predicted. Let's have a look. We did a number of experiments or a number of studies, but in this study from 2019, we looked at gene expression in peripheral blood lymphocytes of two donors. And this is donor 1, this is donor 2. The gene is FDXR, here is XPC. And you see that the relative messenger RNA level, the fold change for this gene is lower for X-rays than for alpha particles. But if you combine both radiation types, then the response is stronger than alpha, and suggesting synergism. Because if it was additive, you would expect the response to look like this. And the same for XPC. But in the lymphocytes of donor 2, the response of mixed beams is exactly as expected. It's additive. You see, it's between alphas and X-rays, both for FDXR and XPC. So are these donors different with respect to their sensitivity? That is maybe biologically, that is genetically determined. Is it genetically determined or not? And in order to answer this question, we ran a study where we collected blood samples from these two donors over a period of nearly 1 year, during season 1 in 3 consecutive weeks, then season 2 again 3 consecutive weeks, and then again season 3 during 3 consecutive weeks. So altogether we repeated the blood draws 9 times, and we exposed cells to alpha particles, X-rays and mixed beams, and analyzed gene expression and chromosomal aberrations. And you see that those responses, both for chromosomal aberrations and for gene expressions, are linear, meaning that we can easily calculate the slope of the dose response, and this allows us to present the results of a dose response in a single number. And we can calculate whether the ratio observed to expected outcome, and if it is synergistic, let's see, it will be red. If it is additive, it will be blue. And here you have the results of the study. This is donor 1, this is donor 2. We have aberrations here. We have gene expression here, different genes, different fold changes. The values that you see here are ratios of observed to expected. And the red values are suggest synergism, blue values suggest additivity. And you see that for donor 1, at least, in season 1, it's kind of red color dominates. Season 2, same. Season 3, blue color dominates. So during season 3, this patient, this donor, and not patient, it's a healthy donor, showed additivity, while in the other seasons, more tendency towards synergism. For donor number 2, similarly, but here the pattern of response with respect to the different seasons is completely different. In season number 3, it was mainly synergism. In other seasons, maybe some additivity. However, if you pull all the ratios together from the different assays, you see that they are all in the yellow-red zone, suggesting synergism. So the response of these individuals to mixed beams is variable, but this variability is more or less stochastic. So we have a case of aleatoric uncertainty. It's impossible to predict at one instance how the donor will react to radiation. Another case, another study that I want to show to you is carried out on patients. It's the radiotherapy patients, and the aim of the study was to test if a patient who receives radiotherapy twice to different locations of the body, if this patient will react in a similar way with respect to his or her normal tissue. So toxicities, will the level of toxicities following radiotherapy be the same or similar following after the first and second radiotherapy? I should say that these radiotherapies were given to tumors in different locations. So we don't talk about re-irradiation of the patient. It's like both therapies hit tissue that was not exposed before. And if there is a good correlation between the first and second, or reactions after the first and second therapy, this would suggest a genetic component. If there is no, it would suggest an environmental component. And you can see that we had 98 patients, and this is a graph that shows you the time between the two radiotherapies for female and for male patients. And it's a collaboration between a Polish hospital and a German hospital. Here you see that you see the location of the first cancer site and the location of the second cancer site. And well, it's just an illustration to show you that these locations are not necessarily the same, although first breast and second breast, then the second breast would be the contralateral breast, which was not exposed to radiation during the first radiotherapy. Here are the results shown for the Polish hospital, for the German hospital, and it's best to focus on the combined results because they are actually very similar. And the first thing that you can see here, if you calculate the odds ratio, so the probability of developing side effects as a function of dose, you see that you get a positive, a significant relationship. The odds ratio is higher than 1, and the confidence limits are higher than 1, suggesting that there is a correlation between the dose that the patient was exposed to and the risk of developing side effects. And this makes, of course, very much sense because we expect the dose to be a strong contributor to side effects. And this positive correlation that we observe is kind of a proof of principle that our approach works well. There was no correlation between sex, surgery, and then let's look at the question whether there is a correlation between the first and second radiotherapy, or the outcome of first and second radiotherapy. And here we calculated something that is called the variance of the random intercept. A low level of variance would suggest that there is no correlation between the first and second, and a high level of variance which suggests that there is a correlation. So a large variance suggests that there is a genetic component. A small variance suggests that there is no genetic component. And in fact, you can see that the level of variance is extremely, extremely small. We also calculated heritability, which I'm not showing you, and it is at the level of 0. So these results clearly demonstrate, for our patient cohort at least, that there is no correlation whatsoever between the reactions of the patients after the first and second radiotherapy, suggesting that there is no genetic component behind the reaction of a patient to radiotherapy. So it's probably lifestyle factors, environment, we don't know. In fact, this result is not surprising because you would expect, or we expect the trait of individual radiosensitivity to be multifactorial or complex. And multifactorial complex traits or polygenic traits are the outcome of interaction of very many loci of low penetrance with the environment. And of course, it would be interesting to be able to assess what is the impact of the contribution of environmental factors and genome to the effect of the trait. But because there is an interaction between the genes that contribute to the trait and environment, like you see here, it is actually not possible to differentiate between the impact of the genome and the impact of the environment. And this is then also, or places us in the position where we say, well, it's actually impossible to predict how somebody will react to radiation. And in publications on this problem of interaction of low penetrance genes with the environment, authors also claim or show or demonstrate that the component of chance also contributes to the trait, which adds another layer of complexity. If it's not just the cooperation between or interaction between the genome and the environment, but also a component of chance, then it is really not possible to predict how somebody will react to radiation, because it's a matter of chance. So what do we conclude from these studies? That chance is a contributor to the individual reaction of somebody to radiation. Obviously, environmental factors contribute to the variability observed, but we don't really know which factors contribute, at least not in the factors that we have observed in our studies. But this must be borne in mind in the quest for biomarkers of individual response to radiation. And in fact, our results suggest that it may be very difficult, if not impossible, to predict, to find a biomarker that will predict how a phenotypically normal person will respond to radiation. And this is my last slide. Thank you very much for your attention. I am happy to answer any questions.
Thank you very much, Dr. Wojcik, for this very interesting talk. So I guess there are some questions, so please.
Yeah, so my question is related, the query is related to your data. You are showing that alpha or gamma, you are not getting much change in the variation in radio sensitivity, if I'm correct to your data. So whether you feel this is ideally this alpha should be basically, gamma should be variation in sensitivities, or X-ray should be more than the alpha. So whether your LET is different, suppose it is comparable to X-ray, that is why you are not getting a difference in sensitivity. Are you
Okay, thank you for the question.
My question is that, so in your data you are showing that alpha and X-ray, we are not getting much variation in the sensitivity, radio sensitivity. So whether your LET of alpha is in range of comparable of gamma, that is why, or X-ray, that is why you are not getting difference.
Okay, thank you for the question. No, the LET of alpha is around 100 keV per micrometer, so it is much higher than for X-rays, of course. We have used, or I showed you the results for mixed beam exposure because I used the results as an, should I say, substitute for a test of individual radio sensitivity. So we regarded the reaction of a donor to mixed beam exposure as a surrogate of a test for radio sensitivity. And in the majority of cases, and overall, if you take the average of all the results, it's obvious that alpha particles and X-rays interact and cause an effect that is higher than expected. But there are seasonal and random variations in the response. And that is my point. If you run a single experiment, you cannot really rely on the results in terms of, is there an interaction, is there no interaction? You can also not really, I would say, predict an individual's sensitivity from a single exposure because there is so much variability caused by environmental factors, I think. That's my message. That's the message I would like to convey to you.
Okay, yeah. What about the radio sensitivity index that we can, if at all, we can use as a clinician, radiation oncologist to treat our patients? Does it also has any environmental effect as a seasonal effect? When I treat in summer, when I treat in winter, has got any difference in that?
I don't know. I would guess yes, because it's a multifactorial trait, and multifactorial trait is the outcome of interaction of the genome with the environment. So I expect yes. Now it's an interesting question. We know there has been investigations on the efficacy of chemotherapy related to circadian rhythm. So it is, now I don't remember whether it is better to start the treatment in the morning or in the afternoon, but there are differences in the outcome. I am not aware of similar investigations for radiotherapy. I don't think it has been looked at, whether it is better to irradiate in the morning or in the afternoon, but I would not be surprised if there would be effects.
Yeah, circadian rhythm, yes, it's accepted to a certain even though we don't really practice it, but it is understandable. But what happens to the seasonal variations? In December it's different. If I treat in the summer, it will be different. In that way, is there any variations?
I don't know. I don't know what factors influence the response. I mean, the two donors that we looked at, I was one of them, and my colleague was another one, and we were healthy all the time. We didn't have COVID. I don't know.
Well, thank you very much, Andre. So to be with us here, and I'd like to thank all the speakers for the great talks and all the questions. And this was a very nice session, and I also thank my co-chair for having me here. And so I'd like to close this session and wish everybody a nice afternoon and a nice evening.
Thanks a lot. Now we are ready for dinner.
Thank you, Chairperson, and thank you, presenters, for wonderful insights. I would like to call upon the stage Dr. J.K. Singh, Director, Sri Sai Institute of Medical Sciences, Patna, to felicitate Dr. Sudhir Chandna, Director, DRDO of Institute of Nuclear Medicine and Allied Sciences, Patna, with a bouquet and memento. Dr. Neil Scott, Professor, Molecular and Cellular Radiobiology, Encore, USA. Dr. J.K. Singh, to felicitate Dr. Sophie Dobash, Professor, Department of Radiation Oncology, School of Medicine, Munich. Dear respected delegates, kindly assemble onto the stage for a group photo.
After the photograph, yes. We want to have photograph. No? I think we have a group photography. So everybody who are present here, if you come on the stage, probably we'll have one photograph.
Everyone, please assemble on the stage for a group photography. And tomorrow, kindly assemble at the auditorium at 8:45 a.m. so that we can proceed with the day 2 sessions. Thank you. After the group photography, dinner is being served.